and verification with Dr. John Ladbury, who's an electrical engineer with the National Institute of Standards and Technology. John?
Good afternoon. My name is John Ladbury. Also here today, we have Galen Kepke, who was our project leader when we got involved in all of this, now retired, and also our group leader who could not make it today, Perry Wilson. I think our side of this really got started. Perry heard about the possibility of doing this project and heard that NTP was interested in doing tests where animals would not be as constrained as they have been in previous tests, and suggested, well, why not use a reverberation chamber for doing these tests? And that's pretty much where we are now, how we got into all this. Real quick, I don't know if being right after lunch is a great position, especially since I would bet 95% of you are mostly interested in the biology, and I'm going to be talking electronics. So I won't be offended if I do see a few winks here and there. What I'll be talking about, start out a quick background of who NIST is. Just give you an idea of why we are involved in this side of the measurements. Quick description of what's some ideal reverberation chamber characteristics, our plan for doing validation verification, our results when we did loaded chamber uniformity, quick analysis. Miles has already done some good coverage of talking about kind of fields and field probes, but I'll give a little bit more information on that. Talk about our empty chamber evaluation and signal quality evaluation, followed by a quick summary. So NIST, we are the National Institute of Standards and Technology. I've taken all of these buzzwords and phrases from various web pages. These are what our organizations like to claim about who we are. For NIST, we are dedicated to measurement science, rigorous traceability, development, and use of standards. Our organization is mainly there to kind of establish standards for trade. We help establish what is the definition of a meter, definition of a second, definition of a kilogram. And, believe it or not, a lot of those may seem like kind of already well done and established. There's a lot of work being done on getting a more accurate kilogram that has nothing to do with actually having a weight that weighs 1 kilogram. They're going to use physical properties that have to deal with forces between things that when there are electrical currents flowing. So those are the sorts of things that NIST does in general. Inside of NIST, we are part of the Communications Technology Lab, which is a fairly new lab dedicated to advances in measurement science, underlying wireless technology. Inside that is the RF Technology Division. We develop theory, metrology, and standards for technologies upon which future of wireless communications depend. And finally, I am in a group called the RF Fields Group. We measure, characterize, and calibrate antennas and radiated fields. Specifically, we are the principal traceability path for RF field probe calibrations in the US. As a group, we have 40 years of experience in reverberation chamber measurements. We've helped establish international standards for calibrating and performing measurements in reverberation chambers. Most of those are for using the chambers for evaluating electronics, electrical systems, anything from automotives, aircraft components. So this was new to actually be involved in something with biological systems. Luckily, the electronics don't change. So the main things that we've learned about characteristics of an ideal reverberation chamber and why they are well suited for this type of test. First step, it doesn't really matter where you place your antennas. There is a little bit sensitivity there because the antennas do have a pattern to them. They have a direction, often they have a direction that they want to radiate. Especially if you see like a dish antenna, horn antennas, They have a specific direction they like to go. So you don't want to take your antenna and point it directly at your test object, because it will tend to get more of the energy, which is why Miles took their antennas and pointed them at the paddles, because it'll stir up the energy, randomize it a little better. It also doesn't matter where you put the probes. As long as you are inside the working volume of that chamber, the working volume is pretty much defined as anywhere maybe quarter to a half wavelength away from the walls, away from the paddles. So you have a very large area inside the chamber for placing probes and placing whatever you're going to be testing. And it doesn't matter whether you put one, close to the wall or in the middle of the chamber. They all get roughly the same sort of measurements. So similarly, it doesn't matter where you place whatever it is you're going to test. And finally, it doesn't really matter how you orient it. It doesn't matter if you place something in like this, place it like this, place it like this. Anywhere around that working volume. For an ideal chamber, you get the same answers. Validations we've been involved in. We actually started out, we visited, it is, before all of this, saw some of their prototype chambers and just looked at some of their, what they were doing. And after that, we did measurements after the 21 chambers were initially installed in Chicago back in December of 2007. In April of 2012, after all the pre-chronic and thermal pilot studies, we came out, did another evaluation, and then roughly around a year after all the chronic tests were done, we came out again. So the chambers were dormant for about a year before we came back and did the evaluations. Start out with just the loaded chamber uniformity. Here we have 2 racks, and as you can see, it's very difficult to get a person into there and do anything, moving anything. Let's see if I can see a mouse. There we go. Right around here, we've got one antenna. What we've done is we've removed one of the cages and replaced it with an antenna. We go through the whole measurement procedure with that antenna in place, just measure the power received off of that antenna with a spectrum analyzer. And then we do that in several different locations, 20, 24, somewhere. Initially we did 20 measurements, just to get an idea how much variability is there around the chamber. It really doesn't matter which chamber we're in, they're all constructed. The uniformity tends to be just dictated by the geometry of the system. For our measurements, we saw a standard deviation in those measurements of about 1.3 dB, which is pretty consistent with the sort of numbers Miles was giving earlier. We saw a range of about 2.5 dB. In general, I don't focus a whole lot on range because you're looking at the difference between maximums, which are very statistically not very stable numbers, and minimums, which are even worse. And so you look at the difference between those, and it's something that can range quite a bit. If we were to redo the test, we could get 2 dB, redo it again, might be able to get 2.8, 2.9, 3 dB. So I like to use standard deviation. It's a better indicator of the average performance of the facility. All the tests we did when we came back and repeated these measurements, they were in the same sort of range. So RF field probes and how we calibrate them. This is not one of the probes we actually use. This is a commercially available probe, but visibly it gives you a good kind of idea of what you have. You have a set of 3 dipoles, each orthogonally oriented. And what you can do is you can create some pick whatever Cartesian coordinate system you like for whatever you're measuring. You can align one of those probes with the x-axis, one of the probes with the y-axis, one of the probes with the z-axis. And You can imagine that if you have a field coming in like this and you have a probe aligned like this, it'll pick up the maximum. If you have something aligned like this, it'll pick up nothing. If you have something aligned like this, it'll pick up nothing. So this is very good for splitting those terms out. For calibrating, we start out where we align. We do the measurements in an anechoic chamber where all the walls are aligned with absorbers, so there are no reflections. It's kind of the opposite of a reverb chamber. And that is designed to simulate a free space environment. If you were in outer space and you could have an antenna that was transmitting energy and have the probe out here measuring, that's kind of the ideal measurement situation for doing these calibrations. So when we calibrate, we'll generate some known signal. If we're interested in just a simple sinusoid, that's what we generate. If we're interested in GSM or CDMA, we will generate a representative signal. One of the challenges here is the more complicated you get, you can't strictly say, I would like a CDMA signal or a GSM signal, because there are slight variations. If you happen to pick one and you decide I'm going to transmit 0, you're going to get a slightly different waveform than if you transmit 0, 1, 1, 1, 0, 1, 0. So there are subtle variations, but for the most part, if you just generate random signals on these modulations, that gives you a good representation. So we generate those signals, we amplify them, we measure how much power is going out to our transmitting antenna, how much power is reflected back off of it. We radiate that signal using using an antenna. The antennas have very nice fixed geometries, which we're able to use to calculate field at a certain distance away, given how much power is going into the antenna. We compute the fields based on that radiated power, and then we measure the probe response. Now, inside this probe, there's a diode. And a diode is a very nonlinear element. And we really like to have linear systems where if we double the field, we'll double the voltage out. There are just no systems out there that are really reliable and are able to do that. So instead of doing a single measurement and we're done, we span over a range of amplitudes. We'll start out at a very low amplitude signal, measure the response, bump up the signal by a dB, repeat. So we cover the entire range that we expect to see during a measurement. Then what we'll do is, once we've got our measurement here, we'll take the probe. It's nice to have these models here. So if we had a signal coming in, initially this one picks up all the signal, this one picks up nothing. Then we go and rotate it. This one picks up nothing. Now we have to calibrate this dipole. So we have to do a set of 3 measurements for each 3-axis probe. For our measurements, our standard uncertainty in the calibration of the probes is about 0.4 of a D, which is about 10% in power. When we visited we checked out their calibration facilities. They do a very similar sort of setup, very similar uncertainties. So we were not worried that things were going to be off by very much. Quick review or explanation of the fields that we might be looking at. There are electric and magnetic fields. Electric fields typically measured using a dipole, part of an E-field probe. You can also use loops, which are used to measure the H field. Initially, when these tests were being set up, there was some speculation. Some people felt that H field was more likely to have some kind of an effect on biological systems. So just in case, each of the chambers were equipped with one E-field probe and one H-field probe. And one of the nice things about reverberation chambers is if you know one, you can predict the other. There's just a simple constant offset, constant multiplier that allows you to go from one to the other. So it really doesn't matter if you use all H field probes, all E field probes, or a mixture. As we mentioned, there's an X, Y, and Z component. The problem with that, having just those individual components, is as you rotate your probe, you start getting different answers. You might start getting more of a pickup on one and not on another. So it's useful to start looking at the sum of the squares of each of those, because that term will be independent of how you orient your probe. As long as you're generating a constant signal, you can rotate your probe, always get the same result. SAR is going to be proportional to the square of the total field, which is the square of the X component plus the square of the Y component plus the square of the Z component. So the basic procedure we went through for evaluating the empty chamber, we had one probe. It's a tiny little thing, about the size of my thumb, probably a little bit smaller. We used this probe and we would place it in each of the test chambers that we evaluated. We also have one of our horn antennas, which we use hooked up to a cable, in this case going up through a bulkhead feed through up in the roof, then goes down to a spectrum analyzer. Use that to measure received power. We can use received power as one way of estimating what the total field is. We can estimate the total field from any individual component. We can mathematically compute what the total field, or we can actually measure X, Y, Z components, square them, and add them together. So we have several different independent ways of estimating the field inside the chamber. For our measurements, the paddles that are in here, we step them a certain discrete number of positions. The tests were run with the paddles moving continuously, but for our probe and our system, the probe tends to respond very, very slowly. So we need to let it sit, let the field stabilize, measure the response, step, let it sit, measure the response over and over 100 times. Here are responses that we measured in all the chambers. Let's see. We've got the chambers listed sequentially. You'll notice there will be a few that are missing. There's no chamber 4, there's no chamber 13, no chamber 15. Those are the sham chambers. There was no reason for us to do measurements in the sham chamber. There were no fields, or at least our field probes are not sensitive enough to pick up what fields would have been in those chambers. So in all of these, the first 3 chambers were CDMA mouse chambers. These were the high, medium, and low values. Then there were the GSM male rats going medium, high, CDMA male rats, high, low, etc. So it's very difficult to just track exactly which of these is which. So we're not going to try. Instead, what we're doing is flattening these out. On the line are the values that it was hoping to hit when these measurements were done. So those were the target values. We also have their probe number 1, E-field probes, the X, Y, and Z axes used to estimate the total field. Their second probe, X, Y, and Z axes, used to estimate the total field. Our probe, X, Y, and Z axes, used to measure the total, estimate the total field, and our received antenna. All of those are plotted along here. To make it a little bit more readable, we normalized everything to the goal. Flatten everything out so we could see things a little bit better. Now, it might seem to make more sense if we are saying that we are defining what the actual test levels are, and we're seeing how good it is, where they rank relative to our measurements, then we would start out, plot our data, and see how it is compares against us. But when we looked, the uncertainties in the calibrations of their probes are very similar to the uncertainties in the calibrations of our probes. The bigger components to the uncertainty are the uniformity of the chamber and the spread we get because we just used 100 paddle positions. If we start using 1,000 or 10,000 paddle positions, we can tighten things up quite a bit, but it takes a factor of 10, factor of 100 times longer to do the tests. So we accepted the small hit that we got by just using 100. And now we'll just say, well, let's use the target value and we'll see how well our measurements match the measurements produced by the NIEHS probes. And overall, if we look at the uncertainties for combining the probe calibration uncertainty, the fact that we're only using 100 paddle positions, and the uniformity of the chamber, we expect a variation of about plus or minus 2 dB for these measurements. Overall, everything seems to line up very nicely within those 2 dB. We've got a couple points that are lying right on the ragged edge, and then we have 2 that are really standing out. They're just a full dB outside of the range, which seemed a little bit odd. They're both green triangles, so that shows that it is H-field probes. And I guess that's the end of my presentation. Okay, or it's not. Okay, so in this case, they were both, X fields on the H field probes. So we wanted to go through and check that out a little bit. If I could advance. There we go. First, what we did was we actually took those probes and measured the total field. We added the sum of the squares to the E field. Now, the error bounds are just a little bit tighter because E field, total E field is the sum of the squares of the 3 components plotted on a log scale. It's essentially equivalent to averaging those 3 terms. So we get a narrower uncertainty on the total field, squared total field, than we do on the individual components. So the bounds are a little tighter. Again, almost everything lies within, now uncertainty bounds are 1.5 dB. Most everything lies nicely. We've got our NIST ET probe, which seems a little bit low on chamber 10, but chamber 6 and chamber 17, again, they seem like they were out of the range. One of the things we did, Miles talked about cumulative distribution functions. This, we take all the data at the 100 probe positions and we sort them. 100% of the data is, say, less than about 7 or 8 dB higher than the mean. Half the data is below the mean, above the mean. Only about 10% of the data is 2, 3 dB below the mean. So that's roughly what these look like. What we want is that all of these curves should overlap. And we had a few that seemed like they were off a little bit. Chamber 5, 16, 17, 6. They're all H-field probes. So we're starting to see that there was an issue. Specifically, we went and looked at probe 6, or chamber 6, which was a high male rat chamber in the GSM range. The H-field probes just seemed like they were off from the rest, and especially up as we started getting higher values, it seemed like suddenly you reached a level and then they jumped up. And After we brought this up, as a fix, it has replaced the H-field probes in the high-level GSM chambers to E-field probes. It just seemed like the H-field probes were malfunctioning. Well, it's the scaling that came out of them mostly was causing problems. So those did get fixed before any of the tests were being run. When we came back in 2012 and did more measurements, now everything falls within the 2 dB guides with maybe a couple of points just barely on the outside. I don't worry too much about things that are very, very close to the edge of the error bar. If you figure that most of the error bars are 95% error bars, you would hope that things actually get close when you start having 20 or more measurements. You should start getting points that are close to your error bounds. If we look at total field, now everything does seem to line up within our 1.5 dB range. So these were the measurements that were taken after the pre-chronic and thermal pilots. We look at the CDFs. Now everything lines up pretty well, but we did take a close look at chamber 6 again, and there are still a few little anomalies in the E-field probes, and it turns out the data acquisition units were being overdriven just a little bit at the very, very high field levels. So they were able to put in a simple voltage divider, brought those back into the range without really affecting sensitivity. All of those problems went away. And actually, and these are pretty rare events anyway, so less than about 10% of the time any of those readings would have been off by just a little bit. They would have had a very small impact on any final measurements. Back when we came back in 2015, again, this was after the system had been dormant for a year. We came back and we found that the zeroing units in 2 of the chambers had failed. So we went back and Miles went, reviewed the data logs, the advantage of being able to keep every piece of data that was taken in these measurements, and found that the monitoring unit in chamber 3 had had issues for about the last 6 months of the test. But the one in chamber 20 had shown no signs of problems. So before we went and did our evaluation, went ahead and for any kind of long-term test like this, you have to be able to replace out parts during the test as they go out. So they have spare parts at any time. They can take one out, put in another one. So they went ahead and replaced the zeroing unit in chamber 20, but since we know that there were problems in chamber 3, we went ahead and tested it with the malfunctioning zeroing unit in place to see how much of an effect that would have. And you can see there was definitely one point that was actually more than 7 dB off, caused by those zeroing units. A few other points that were a little bit out. But when we did measurements of just the total field, all of those points had a negligible impact on the true total field, which is what the fields were being calculated from for all of the tests. So even though chamber 3 did have a malfunctioning unit, the readings were maybe a little bit low, but there's nothing that really stands out, so should have had a negligible impact on any of the measurements. Went back and plotted a CDF. Once again, there was just a single axis from chamber 3 that showed any problems. And it's very clear from this that there was an issue. But all the rest seemed to be in working order, even well more than a year after the end of after they've been sitting dormant for a year. Finally, we look at a little signal distortion. This is a little bit tricky. There are, there may be distortion effects that could cause a cell phone not to communicate properly with its base station, but would be most likely a very low level effect. Whether something like that could have any effect on a biological system when most of these anomalies are a factor of 100, a factor of 1,000 below the main signals, I can't really answer, but I can say that any of these effects that might have been there would have been very low. So the type of standards, communication standards, they say you can have your signal that you want within the band up to here. By the time we get maybe 100 kilohertz outside of the band of interest, we have to be a factor of 1,000 down below that. And then we get a little bit farther out, we've got to be at least a factor of 10,000 to 100,000 below that center signal. So we just looked at a few characteristics. One of the things that is important to check whenever you have an amplifier, which is a nonlinear element in the system, there's always a possibility that you're going to generate extra signals at multiples of your main signal. So for a 900 megahertz signal, it's possible that you might see something at 1800. And there is a very low level signal here at 800. But once again, if this is at a 0, this is about a factor of 1,000,000 lower. So very, very low signal. We are almost down to our ability to measure anything. This is the ability of our sensitive spectrum analyzer to even measure these signals. Similarly, for a 1900 megahertz signal, if we look at 3800 signal, there's a factor of 1,000,000, so almost a factor of 10 million below the fundamental signal. The other thing that we look at is these are the other guides where we have to be 30 or 40 dB below the fundamental within these bounds. So those are very close. There might be something very ragged, just barely above. That might be enough that if you own one cellular company and your neighbor is just peeking over, it's probably not going to cause you a problem, but if it does, you might speak up. But most of the time, that's never going to cause a problem, even on a communication system. The CDMA measurements, we did not see anything in the harmonic content. We couldn't see anything sticking up above our noise floor. So we'll just put the two frequencies on the same chart here. At 900 megahertz, things are falling down well within the communication spec. Both of these fall well within the communication spec. So no problems. Let me go back. These signals, CDMA signals, they have a certain width to them. When you look at them in the time domain, as Miles said, they look just like noise. They basically look like Gaussian noise type signals, always on. That signal is always on. When you move on to a GSM signal, those signals are impulsive. And so they're on for 1 cycle, off for 8 cycles. On for 1 cycle, off for 8. During the time when this one is on, the other chambers are not. And we wanted to see what happens when we're looking in chamber 6 when it's not supposed to be picking up anything. So we're actually transmitting GSM into one of the other chambers on these lower signals. And a little bit of that signal bleeds through the switches back into chamber 6. Typical switches, RF switches, will have attenuation of 20 dB or so. So the fact that these signals are down 40 dB, factor of 10,000 or more, the bleed through is very, very small. Very, very low level type bleed through. the fact that we have different colors here, we took measurements at different paddle positions just to get an idea of how those ranged as the paddles turned. So to give a basic summary of what we found, loaded chamber uniformity had a standard deviation of about 1.3 dB throughout all the tests that we've done. We did see some unusual probe behavior at various points in the tests, but those were fixed very quickly. For chronic tests, we did not see that any problems were there for the chronic tests, and anything that might have been any problem would have had a negligible impact on the signal levels during the thermal and pre-chronic tests. Again, after the chronic tests, we did notice that 2 of the zeroing units had failed, one of which was replaced. The other we evaluated with the malfunctioning unit in place and had a very, very small impact on the estimated fields. Signal quality that we measured was within standard parameters for the communications standards that are available for CDMA and GSM. So other than the things we pointed out, which did get fixed before any of the major tests, there were no problems that we could identify with these systems. So that concludes what I have to say. If there are any questions?
Questions? Dr. Lin?
Okay, thank you for a nice summary. You started off by saying that the reverberation chamber is a very robust system. Really doesn't matter what you do, you're going to get the same answer. Is that correct?
Within your uncertainty bounds, yes.
Okay.
So
That being the case, let me ask the next question: that who designed the particular RC that was used for the NTP study?
well, I mean, there have been general designs for reverberation chambers that have been out for quite a while, and we've had people that have gone out and taken samples regardless of how they've been made. And with a few exceptions where there are obvious things that someone has done wrong, they consistently work very well. For these specific chambers, they were designed by the folks at NIEHS.
Okay. So you also said that you guys, NIST, came up with a particular reverberation chamber. Then, like you said, then the particular one was designed by NIEHS in Zurich. That's what you're saying, right?
I believe so, yes.
So given that it is a very robust system, very little can go wrong, why not design and build it in Chicago instead of doing it in Zurich?
I'm not sure I'm the person to answer that question. If anybody else wants to take that,
Dr. Booker will make a comment.
I'm trying to remember all the things that were going on. The whole system is put together through contractual arrangements with different organizations, and I think we were taking advantage of the capabilities of the contracts that we had in place at that time. I don't think we had the capability to put the chambers together in Chicago. Yeah, as you saw earlier, all of the plumbing and electronics and everything that were assembled were done in Zurich, and you saw the care with which that was taken.
and I can also add that there are many organizations you can buy commercial reverberation chambers that are mostly designed for doing electronics testing. At the time that this was going in, I don't know of any commercial chambers that were available that were considering constant cleaning effects, anything for associated with the life sciences, dealing with the light levels, dealing with all of the specifics that go into the animal care, and just details like light, ventilation. The standards that are required for animal studies are much, much higher than you have to for testing aircraft components.
Any other questions from the panel? Yes.
Mark Klein, Wake Forest. Did I understand correctly that that no measurements were made in the control chambers?
That is correct.
I mean, we had field probes inside the control chambers, so we did record the noise level of our measurement system throughout the study.
Further questions? If not, we will move on. Thank you, Dr. Lambert.
Thank you.
So Dr. Mike White will return to the podium and give us an update on the thermal pilot studies, reproduction studies in rats, and exposure level selections for NTP studies.
All right, good afternoon. It seems like we're finally at the interface between the technology and the biology. So this talk is really the first of the biology that we'll see from these studies. And so in my talk, I'll give a summary of the thermal pilot studies, which is the first time that animals were actually placed into these reverberation chambers. So this is a slide that I showed before. So basically what we'll be talking about in this talk is these 5-day thermal pilot studies. And to go over my talk, the first thing we'll talk about is the goals of the reasons why we did these 5-day thermal pilot studies. We'll talk about the design of the studies. We'll go through the rat results and then the mouse results, and then we'll sum it up, and we'll have a discussion about everything that we talked about this morning. So the goal of these 5-day thermal pilot studies was to evaluate a wide range of specific absorption rates, or SARs, to determine like a threshold, where the threshold of potential thermal effects following exposure to RFR. As Dr. Capstick mentioned before, there's some issues as far as the size of the animal, which is related to the mass ratio of surface area and the ability of larger animals to dump heat into the environment. So basically what we wanted to do is to investigate the impact of animal size and pregnancy status on the exposure levels or the response to heat. So basically the goal was to determine what power levels might be excessive as we moved forward to select exposures for the subsequent studies, the 28-day studies as well as the 2-year studies. So the goal here was to identify power levels that were below where the animals could thermoregulate, and specifically we set a pretty widely accepted standard of 1 degree C increase as being defined as an excess increase in body temperature. So again, we also wanted to determine, while we had these studies going on, any potential effects of RFR on the pregnancy status in the rats. So these studies were conducted in a series of 10 different studies. The first 6 studies were done in rats. These were young, aged, and pregnant rats. These were exposed to either GSM or CDMA. So there were 3 studies of each of these 3 of the GSM studies, 3 of the CDMA studies. And then we also had 4 studies that were done in mice. We had young and aged mice that were exposed to either GSM or CDMA RFR. For each of these groups, there were 5 animals per sex, per exposure group. And as I mentioned before, the daily exposures in this case were up to 12 watts per kilogram. And as I mentioned, it was the exposures themselves were for 9 hours and 10 minutes a day. This was through 10 minutes on, 10 minutes off cycling. So Again, as we've discussed all morning, rats were exposed to the 900 megahertz signal. Mice were exposed to the 1900 megahertz signal. In the pregnant animals, the exposures for the 5 days were over the course of gestation day 10 through gestation day 15. Now, as you've seen all morning, we have 21 chambers. There's a low, medium, and high chamber for each of the modulations. And so, as I mentioned, we're looking at 4, 6, 8, 10, 12. That's more than 3 exposures. So because we wanted to look at an expanded dose response range, we had to conduct these series of experiments in multiple cohorts. We could only do 3 exposures at a time. So the way that we conducted these, where we did the 0, 4, 6, and 8 together, and then we did the 0 and 10 and 12 together. And I'll show you as we move through the data how that's presented. So we collected body temperatures through implanted microchips, which you can see here at the bottom of the slide. This is obviously not to scale because this is huge, but the size of these are about 14 millimeters in length, about 2 millimeters in width. These were implanted subcutaneously into the animals. And with this wand or this transponder on the right, the staff was able to go in as soon as the system was shut down to measure temperature as quickly as possible. I believe Miles showed a graph of the decrease of temperature, or the loss of the temperature. So it was imperative that we take these readings as quickly as possible after the exposure was stopped, so we could make sure that there was no time for the animal to dissipate that heat. So again, the staff was able to, at the immediate cessation of the exposures, to run in with these wands and blip over the animal to record the temperature data. The temperature was collected at 3 time points each day on days 1, 3, and 5. Again, this was collected right after the cessation of the exposures. It was also collected on days 2 and 4 during the period of animal husbandry, after the animals had about a half hour to an hour or so of rest from the exposure. Today we'll just be presenting the collection from those 3 days, and all of the data can be found. The reference is here on the bottom. This was White et al. That was recently accepted for publication by Bioelectromagnetics. So dams from the control group and 10 and 12 watts per kilogram were also evaluated by cesarean section to evaluate potential effects on the status of the pregnancy in these animals as well. So, as previously mentioned, males and females have different weights. I think Miles showed the graph twice that displayed this quite nicely, but here you can see that the weights in males and females, males are quite heavier compared to the females in all of these rat groups. You see the mice, males and females are approximately the same weights. We could also see that the weights between that were used in the GSM study were very similar to the CDMA study when you look here. However, for the aged rats, there was a bit of a difference. I believe that the males were about 7% heavier and the females were about 14% heavier in the GSM study compared to the CDMA study. So when we talk about age of the animals, it's really not so much that the age that we're talking about, is more the size. So we use an older animal just because the size is larger. So you'll see that we refer to young animals and old animals, but really what we're talking about is smaller animals and larger animals. So as I mentioned, there were 6 rat studies. As I mentioned, young males and females for GSM. So each of the permutations of the exposures are here. I'm not going to go through each of these studies individually. I'm going to present this data collectively so you can get a feel for what type of response is observed following the RF exposure. So we'll look first at the body temperature in young male rats. So just to orient you to this data, as I mentioned, these studies were done in 2 cohorts. So you see here along the x-axis is the watts per kilogram that the animals were exposed to. On the y-axis is the body temperature that was recorded. And in this particular segment that we're showing here, this is the control for the 4, 6, and 8 watt per kilogram exposures. And we see that there's a statistically significant increase at the 8 watt per kilogram exposures in GSM. When we look at the second cohort, we add in the data from the 10 and 12 watts per kilogram. We have a separate control here that was done at the same time that these animals were done, which overlaps, so you can't really see it on this particular graph. But this control refers to the exposures at 10 and 12 watts per kilogram. And as you can see, at 10 and 12 watts per kilogram, we also see that there was an increase in body temperature. When we look at CDMA, we see, again, very similar response. Here is the control for the first cohort, with the control for the second cohort above it. With CDMA, we didn't see the effect at 8 watts per kilogram, but we did see a statistically significant effect at both the 10 and 12 watts per kilogram exposures. So again, this is for male rats. When we looked at the data for female rats, as is indicated here, there was no significant differences in the female rats. This was only observed in the male rats that were of smaller size. These were the young male rats. And this data, I should mention here that this data, as I mentioned, there were 3 time points per day. This data represents an average of each of those 9 time points on those 3 days to give us a better idea instead of looking at the specific time course of what happened. So this allows us to then compare specifically the SAR versus the body temperature. Individual data is available in the manuscript if you want to take a look at the individual data. So when we look at the aged male rats, we saw that exposure to 10 and 12 watts per kilogram, there was not just decreased survival, but all of the animals in the 10 and 12 watt per kilogram exposures were found dead after the first day. And the female rats exposed to the 12 watts per kilogram, temperatures were recorded that exceeded 3 degrees centigrade above the controls, and these exposures were then stopped because this was considered to be an excessive exposure. We see similar results for the CDMA modulation. We see that all male rats at 12 watts per kilogram died during the first day of exposure, and at 10 watts per kilogram, these exposures needed to be stopped because we saw an excessive increase in temperature that exceeded 3 degrees C. There seems to be a slight difference here between GSM and CDMA modulation, but as I pointed out in the table before, this may just be a function of the fact that the animals for GSM were slightly heavier than the animals for CDMA. And it could just be related to the size of the animal, that there is some slight differences here between the GSM and CDMA modulations. To look at the graphs, what we see is a more robust effect in the aged male rats than what we saw in the young male rats. Again, we see as we increase SAR that there is an increase in body temperature, that we see statistically significant increases at 6 watts per kilogram and above for the older animals. The increases at 6 watts per kilogram were below the 1 degree C defined cutoff that was considered excessive thermal increase. But the exposures at 8 watts per kilogram and above, there were increases that were seen that were above the 1 degree C cutoff. And this occurred for both the GSM and CDMA exposures. In the aged and pregnant female rats, we saw similar results between these two groups. The SAR-dependent increase in body temperature was observed following exposure at 6 watts per kilogram for both GSM and CDMA. Again, we saw something similar as we saw in the males. We saw that the increases in temperature exceeded 1 degree C at 8 watts per kilogram and higher in these exposures. And we saw an increase in the number of resorptions when we looked at the cesarean sections. We observed this only at the 12 watts per kilogram and only in the GSM study, not in the CDMA study. There were no treatment-related effects, however, on the number of live dead pups, the number of corpoludia, or the number of implantation sites, or mean fetal weight. So just to summarize what we saw in the rat studies, when we look at exposures at 10 and 12 watts per kilogram, these were excessive exposures. They caused excessive increases in body temperature in pregnant females and aged male rats. We saw increased mortality at these exposures. And when we look at 8 watts per kilogram, we see again that there were several increases that exceeded this 1 degree C set point for what was considered to be an excessive increase in body temperature in both the pregnant females as well as the aged males and female rats. For 6 watts per kilogram, we did see in the rats that there was some temperature effects. These were somewhat minor effects that didn't exceed the 1 degree C, so there was some response, but it did fall below what we were considering our upper thermal limit for change. So overall, what we observed was that the effects were more robust in the aged or larger than the younger rats. And it seems to be that the male rats may have been more responsive than the female rats. And again, this may be related to the size issue. So switching gears, we'll go to the mouse studies. We saw somewhat different results for the mice. Again, there were 4 studies here looking at the young and old males and females exposed to GSM and CDMA. Since we were not planning on doing in utero exposures in the mice, we did not look at pregnant mice. When we look at the data, looking at the worst case scenario, we're looking at the larger animals versus the smaller animals. We did not see any significant differences. We did see some statistically significant increases that were sporadic. As you can see here on the right-hand side, there was a significant increase at the 8 watts per kilogram. But when we look at 10 and 12 watts per kilogram, we did not see any significant difference. And this is what we observed across the board in aged mice as well as the young mice, regardless of the modulation that was tested. So the summary for mice, again, we did see some changes that were sporadic in some body temperature changes that were sporadic in nature. These increases were not considered to be exposure related. There were, again, no differences between males and females and no differences between the GSM or CDMA exposures in these animals. And these data suggest that these animals perhaps could have withstood higher SARs. And we took that into account when we selected exposures for the subsequent studies. So just to kind of touch on that, this is, I believe, my last slide. Just to summarize what we saw from these thermal pilot effects, we saw that there were lethal effects at 10 and 12 watts per kilogram. These were not indicated for future studies given the results that we saw. Again, there was increased resorptions as well at the 12 watts per kilogram. And based on this data, we selected 3, 6, and 9 watts per kilogram for the 28-day studies. We'll be talking about these tomorrow, but we did know that there were some issues at 8 watts per kilogram, and when we selected the exposures for this study, we wanted to make sure that we did actually see some effects. We wanted to push these exposures a little bit in the rats to make sure that we were exposing them to high enough levels, and this also gave us some more information as we move forward to select exposures for the 2-year studies. In the mice, as I mentioned, there were no thermal effects that were associated with SARs up to 12 watts per kilogram, regardless of the age, the sex, or the modulation. And since we didn't see any effects up to 12 watts per kilogram, we considered increasing these exposures for the 28-day studies higher than this. We selected 15 watts per kilogram as the highest exposure in these 28-day studies, primarily because we wanted to go higher, but we were limited also in the power capacity that the system was able to deliver high exposures of RF to the animals. And so based on the limitations of the system and the fact that 15 watts per kilogram was actually quite high, we selected 15 watts per kilogram for those exposures. We'll talk about the 28-day and the 2-year studies over the next 2 days. So that's all I have for the thermal pilot studies. I'll take any questions.
Yes, Dr. Adler.
Rick Adler, GlaxoSmithKline. Thanks very much for the quick overview. Appreciate it. Did you measure body temperatures at night or only during the light cycle?
It was only during the day.
Okay, so you recognize that rats and mice are metabolically much more active at night. Did you do any other background check on thermal variation, diurnal variation of rodents in a 24-hour cycle?
We didn't measure anything.
And you mentioned the decay rate. So what was the time required for a worker to enter the chamber or to remove the racks from the chamber,
So
get the animals out and measure body temperatures versus the decay rate?
Right, that's actually a very good question because it was such an important issue. So if you could kind of picture, we've seen a number of images of the chambers. We've seen them loaded with 2 racks. In this case, there was very few animals. There was only 5 animals per group, so there was only 1 rack in each of these chambers. This allowed the staff to go in and actually get inside the chamber and actually measure these. So that allowed us to get in much quicker. I believe for the thermal pilot study, all the measurements were made within 3 minutes. And it was done actually because there's a number, I mean, you've seen the facility, there's actually a number of chambers that all needed to be measured at the same time. So if we were doing GSM, all of the GSM chambers for rats needed to be measured at the same time. So there was 8 chambers if we're doing controls as well. And so everything was done with a concerted effort. As soon as the system shut down, there was actually a staff member at each of the chamber doors. So immediately when the system was shut down, they were able to enter the chamber and independently measure each chamber simultaneously. And so we did get that time down to, I think it was between 2 and 3 minutes that we were able to take that measurement.
And sorry, did you independently measure the decay rate of an animal whose body temperature was raised?
No, we didn't actually. We did some early studies early on in some animals just to make sure that the chips themselves were working, and we actually did do some heat lamp experiments to actually look at the decay over 10 minutes. It wasn't anything that we published, but we could determine that by 10 minutes, it seems like everything was back to normal for both of the animals that we looked at.
And last question, sorry, it's a series of questions on the same theme though. Did you measure any other physiologic parameters in those animals besides body temperature, for example, respiratory rate, or do any clinical pathology to look for acidosis?
No, and these studies were really just pilot studies in that sense. Again, this was the first time that we actually put animals into these chambers and were exposing them. So the goal here was really just to look at gross effects, to look at the body temperature, any changes in body weight, survival, pregnancy status. We did not look at any histopath or any other endpoints like that. We figured that we would build that into the 28-day studies. And there's, again, we'll revisit this tomorrow and the next day, but we actually did temperature measurements in the 28-day studies as well.
Difference between 5-day and 28-day is rodents acclimate quite a bit to whatever challenges you give them. So we like to see these measures in the pharmaceutical industry often done in a 5-day study because the animals have not reached a steady state. You get to understand what additional systems are perturbed by your external influence. Thanks.
Dr. Felter?
Yeah, Susan Felter, can you tell us a little bit more about why 10 minutes on and off was chosen, and is there anything known about, did you do any experiments with longer exposures than 10 minutes at a time?
No, it was just the 10 minutes on, 10 minutes off. And I believe, someone else can correct me who was involved in earlier designs of the study, but I believe that the 10 minutes allowed the animals a long enough rest period. But I don't know that we had actually looked at whether it was 15 minutes would be different than 10 minutes or whether an hour and an hour. I think the 10 minute I think is a good short enough exposure that you're not exposing them for too long and affecting the thermal regulation and giving them enough time to thermoregulate. But there were several reasons, as I mentioned previously and as Dr. Kuster mentioned earlier, as far as the intermittent exposure, that was something at the time that was considered to maybe be important to there being some type of response to the RF exposure. But the 10 minutes itself, specifically for 10 minutes, that was somewhat arbitrary.
Other questions from the panel? Oh, yes, go ahead.
So regarding the temperature sensors, so they are implanted, and I saw you have a reader. So how do these two devices communicate, like the implanted temperature sensor and the reader?
Let me see if I can pull that slide back up.
Yes,
All
here.
right, so the chip itself was planted subcutaneously, and it was recording. So when the system was shut down, the staff went in with this device here and then was able to, I believe that this button here is what you press, one of these buttons, and it actually is a transponder, and so the information was transferred from the chip to here, and then what was able to be done was once all that data was collected, it was then dumped into a system where we could then manipulate that data.
So is this like an NFC or an RFID, or what's the frequency of communication? Because it's wireless, so I'm wondering whether it's adding app.
That I'm not sure, but I would have to go back and take a look.
Okay.
Although Miles might know because he's been working with some of this.
Yeah, it's RFID type
Okay.
system, so it's magnetic field. I think it's 120 kilohertz, something like that. So there's a little ferrite antenna, both in the sensor and in the wand. So the reader energizes briefly
Yes.
the transponder.
Same. All right. Thank you.
I don't believe that there was any interference between the microchip and the RF that was being exposed as well.
Dr. Lin?
Follow-up question, perhaps looking ahead a little bit, if you were going to do this again, what other temperatures would you monitor instead of just the subcutaneous temperature using a fairly large implanted sensor?
Well, it's interesting that you mentioned that. We are looking at some follow-up studies, and we are looking at some data loggers that actually are able to collect this information in real time, and that way it gets dumped at the end of the exposures, and so it would collect, instead of waiting till the animal was, the exposure was stopped, it would be collecting it in real time. So I think that will give us a better idea of what might be going on with temperature, and I think if we could have used those here, I think that would have been a better system.
Any considerations of location where you might implant the sensor?
No, I mean, when we looked at these particular microchips, the vendor had looked at a comparison between the subcutaneous temperature and the core body temperature, and they showed quite nicely that they matched up quite well. We didn't do any of that work ourselves to look at our situation, but the vendor had actually done quite a bit of that work.
Dr. Barnes?
Did you by any chance look to see if there's any distortion of your fields with your sensor under the skin? Did that by any chance concentrate the fields at all?
I'll defer to Niels on that.
Yes, so we have evaluated the sensors with respect to local heating caused by the presence of the sensor, and also regarding the the interference of the exposure to the sensor field. But as mentioned, the sensor have been read always after, and we could exclude that there is any interference for measurements after.
So I understand that was part of your modeling then, so that it included those dielectric constants and conductivities.
Yeah, it was part of the modeling and was part of the experimental validation.
Fine.
Okay, thank you. Other questions? Yes, go ahead.
Yeah, Matthias Rinke, Germany. Maybe I have missed it. I assume these studies were done on the same strains of rats and mice?
Yes. Yeah, so the rats were the HSD, the same strain that was used in the 28-day and 2-year study. Same with the mice. They were B6C3F1/N mice.
Okay, other questions from the audience? Yes.
Okay. So, I mean, as per, this is Gonçalo Gamboa, Food and Drug Administration. As per what I remember in the models this morning, there was a suggestion that the energy absorbed was substantially more in the core part of the animal than in the outside. So, given that you're probing the external temperature of the animal, what are perhaps could you be missing some important information about the temperature of the main organs?
It's possible, and I think we've had a lot of discussion. It's interesting that you bring up the male organs, and that's when we look at temperature. Obviously the testes are one of the more sensitive tissues, and I think it was kind of interesting that while we are seeing some temperature effects, we didn't in any of these studies really see any of the testicular effects that using that as a surrogate. But yeah, we could have. This is just one way of measuring the temperature.
Sorry, I did not mean male organs, main.
In the main organs. Yes, that is a good point, yes.
But actually, it turned out to be a very important question. Actually, from the dosimetry that was shown early on, in fact, the hottest spot is near the tail of the animals, as you might recall. So in terms of selecting sites for temperature monitoring, you might consider the anatomy a little more as to where to implant it rather than where it is convenient. Another suggestion, perhaps, is that if I understand that there's very little ongoing physiological monitoring what's going on, but if one was going to do that, instead of using something like this or implant ones, depending on what stage of the technology we happen to be in, maybe that's better. Otherwise, the tympanic membrane could serve a tympanic temperature sensor could serve as a very quick read of what the temperature might be immediately following exposure.
That's a good comment, yes.
Go ahead, Miles.
Yeah, Miles Capstick. So I think, what we did was modeling of the SAR distribution, and the SAR distribution is not necessarily directly related to the temperature distribution in the animal. And in other studies, we have done temperature modeling as well. But I don't remember the full specifics of it, but I just want to point out that there isn't necessarily a completely direct relationship between the two.
And I think it's important to also keep in mind, what we're looking at here is more of an overall reflection of what body temperature is, but we don't know what's going on at a localized level. And so when we look at RF and we look at the potential role of impact of heat, I think it's very difficult to get at what might be happening on a localized level. It's easier for us to record what's happening on a whole body level, but that might not necessarily be reflective of what's going on a smaller scale.
Dr. Barnes.
I just happened to think, did you by any chance think of looking at changes in temperature with an IR camera to verify some of your calculations? Seems to me that might be an interesting way of checking to see whether you get the same numbers. It just occurred to me during the course of this talk. It really should have been a question earlier.
Yeah, so people have tried to use thermal cameras in dosimetry, even for phantoms. The sensitivity is just not there, which is needed. And to look at the surface of a living animal would be not very conclusive, the results. But on the other point, when these experiments were not performed in order to look at maximum temperature increase for any of the tissues, my understanding was that they were only conducted to understand what the regulatory system of the animal is capable to deal with.
Okay, thank you. We will take about a 15-minute break and resume promptly at 3:15, in which we will open for public comment period. Good afternoon. We will get started here shortly. Could I have the next slide for the public comment? So we are now going to start with our public comments. I want to first acknowledge and thank all of the people who have submitted written comments. They are shown here on the screen. That's the first slide. You'll see the names there. Could I have the next slide, please? Next slide, please. Okay, there's this second page of people who have submitted written comments. And I believe this is the last page, perhaps. We'll see. Maybe, oh, there's 4 pages. Okay, so that's. And let's check for one more. Next slide, if there is. Okay, I think perhaps that is it. Oh yeah, there's a couple more there. So we have 5 people who are presenting today, providing public comments. I would remind the speakers that we have a limit of 5 minutes for time. On the podium, you will see a little clock there. It will give you basically a 1-minute warning, and I would ask that we not run longer than 6 minutes. So at the 5-minute time, you'll see that you've got a minute to finish up. Also, I just want to remind those who are presenting comments that this is one of several public comment periods, and this really should be focused on the exposure systems comments today, because that is what the panel is dealing with. So with that, I welcome, and I'd ask that the speakers go in the order that I have here. I think you've seen that. Please introduce yourself and where you're from. Theodora Scurato. Thank you.
First, let me just make sure I have this right. Okay. Can you hear me? Yes. Okay. Hi, I'm Theodora Scurato, and I'm going to talk about the unique vulnerability of children, the ever-increasing combined exposures to the public, and the reason the study is so important in that it exposed all tissues to radio frequency radiation. The carefully controlled exposures used in the National Toxicology Program study make it unlike any other, and I've understood the design was intended to test the assumption that non-thermal levels of radio frequency were safe. This decades-old assumption is the basis for our U.S. federal guidelines on cell phones and wireless exposures. We know that children are exquisitely vulnerable to radio frequency. They have thinner skulls, smaller heads, a shorter distance from the skull to brain centers, smaller ears, so if they put the phone up to their ear, it's going to be a shorter distance to their brain, and different dielectric properties, higher liquid content on their brain. And their brains and organs are less myelinated and still developing. So in this video that was running earlier, it was showing some easily findable videos from the internet about children using wireless devices. And here's a virtual assistant, which a child is cuddling, and she's actually singing a song to it. The average radio frequency energy disposition for children exposed to mobile phone radiation is twice as high in the brain comparatively, and 10 times higher in the bone marrow of the skull compared with mobile phone use by adults. And this didn't completely come up, but there was a question about research looking at children's exposure. Published research modeling children's exposure confirms that children's heads and brains are proportionally more exposed compared to an adult, and this is some modeling that shows the higher intensities penetrating deeper into their brains. For some reason the slide didn't come up all the way, everything that was on here. But you can see the darker colors representing the higher intensity SARS, peak SARS, and the lighter colors, there's a 34-year-old, a 6-year-old, and a 3-year-old. Oh, I see. This is what happened. Okay. And what I wanted to talk about was that the way people are using phones and devices is unlike we could have imagined, I think, a decade ago. I was just out and I saw this, so I put this up here. I pulled the picture from the internet, but a woman was walking. She was talking on her cell phone with her baby in this position, and she was kissing her baby. And the bottom 1/3 of the phone was touching the baby's head, and she was taking a stroll, as I was taking a stroll, actually in a forest in Virginia, where the signal was quite low, which means the power was likely quite high coming out of the phone. I'm not sure she was aware that she was exposing her baby to radiation. It was touching the baby's skull that is being studied here in our top research laboratory. When we consider current exposures to radio frequency, people's exposures is no longer just from a cell phone. It's additive because people are using multiple devices at one time, And as an example of many studies, this study showed modeling of the SAR from a cell phone to the head, in addition to the exposure, the SAR from a wireless laptop. And they found an up to 20% SAR increase with the combined exposures. So the first image is just the cell phone to the head, the second was just the Wi-Fi laptop, and the third image was the SAR increased by 20%. During his presentation, Dr. Kapsik talked about how much they needed to develop special water systems to avoid overexposure to the animals, because if they used metal straws, they might be overexposed. And also with people, metal near the body, as well as inside the body, will increase and alter SAR in ways we do not expect, in unexpected ways. Published research has found local SAR enhancement from eyeglasses with metal frames, metal jewelry, piercings, storing phones next to metal objects like keychains. Metal implants can also enhance the SAR in the tissue close to the metal implant, skull plates. Metal in your jaw, like someone I know recently who got metal in their jaw. Hip replacements. Also, there's the issue of metal outside your body in close vicinity, such as when you're in a vehicle. There's a study that'll be on my list at the end with all my studies that are part of this, that it leads to reflections and wave changes inside the vehicle. There was a study that found that even passengers without cell phones had increased SAR exposures because of the nearby passenger with a phone. And now I do have a teenager, and there are many children in a vehicle with many operating devices on at once, more than one per child sometimes. So all of these issues are critically important, and they're not addressed by our current FCC limits. This is modeling with dosimetry done to simulate the exposure of virtual reality, now brought into schools nationwide and across the world, actually. Children are given cardboard with cell phones inside. They hold it up to their eyes. I certainly have seen children with glasses, which would be metal framed glasses, holding these up. It's a smartphone inside of the cardboard. And this is modeling done to show the SAR into the eyes and brain of a 6-year-old child. What are the effects on the eyes from these devices? The public is unaware of the fact that their phones or their wireless devices emit radiation, much less that there are actually health concerns from these exposures. The use of wireless devices and exposures are only increasing. Both our near field and far field exposures. And if you take a look, this was an image recently sent to me of a person who has this small cell right in front of their home. This is their balcony, you can see there. And then the second image is modeling of peak exposures from a Wi-Fi laptop, which let me see if I can, I don't know how to use them. Okay. Which is of course slowed down by several billion times, but laptops have fine print warnings on them stating that, and most laptops are at 20 centimeters, that's about 8 inches, that they are not supposed to be on the body in order not to exceed FCC limits. And yet, college students have these devices on their laps all the time. So those are my comments about combined exposures, children's exposures, and how important it is that this study looked at exposure to all the tissues, all the organs of the bodies of the animals. Thank you very much.
Thank you.
Good afternoon. My name is Olga Naidenko, and I am here talking on behalf of Environmental Working Group. And I don't have a slides. I think for those of us who have been working on this topic for a few years, we can probably talk it off in the middle of the night, just wake up and deliver comments on the spot. Environmental Working Group is a non-profit organization based in Washington, D.C. This year we celebrate our 25th anniversary. Over the course of this time, EWG has worked on everything from agricultural policy to water pollution. And sometimes, I wish we haven't, for the last 10 years, we have worked on the issue of EMF. Anybody who has, like me, been trained in classic chemical toxicology, animal studies, pesticide medications, EMF is mind-bogglingly complex and nothing is as we expect. So our first comment on this portion, on this panel is, I am here to really express EWG support for NTP and for NIEHS for having embarked on the study, for Dr. Melnick for having pushed it ahead. This research is absolutely essential and we hope it will continue, and I will make some remarks exactly where we think are important areas to address in future research. Having worked on this for a few years, I can't help but make an anecdote. In September 2009, there was a Senate hearing, and Dr. Booker was sitting on one end of the table, and I was sitting on the other one. And that's when we first saw those photos of the prototype reverberation chambers. And in 2009, it felt like, oh my gosh, it will take forever for the study to be completed. Can we really wait that long? We have to take action now. Anyway, I am slowly learning that sometimes it's okay to wait, and the wait was worth it. So again, on behalf of EWG, wanted to say it is amazing that this, I would like to say, first part of hopefully ongoing NTP research on this topic has been completed. Our comments, EWG comments, are of course on the record. They're probably in the packet for everybody around this table. Our top message on the exposure study is EWG believes that this research is relevant to people. It is relevant to people for exposures that some people are facing today. We realize, of course, and that's another mind-boggling thing about EMF research, is the study is done on 2G technology. 3 and 4G devices are on the market today. 5G is being rolled out with FCC support as we speak. So even though NTP has nothing to do with risk assessment, but for the rest of us who do, that is the real challenge. And EWG position is that we have to go with the science in hand. We at EWG all use our cell phones. I am so pleased and grateful that NTP is providing free guest Wi-Fi. We could not do our job without it. And yet we take research like the NTP and the Ramazzini study very seriously. So no scientist wants to go beyond the range of data. And yet again, in our exciting EMF field, we have to start with 2G data and then answer the questions which I'm sure everybody working on this issue receives on a regular basis. So what does it mean for us today? And EWG believes that the data are very relevant. If I have another minute, we would like to make a comment about where we think is the next step for the exposure studies. From the physics part, the reverberation chambers are amazing. That is a true breakthrough. And yet from the biology part, we have a lot of catching up to do. And I would like to, for example, mention the study that probably everybody here knows. It's a study from the National Institute on Drug Abuse, or whatever they're called nowadays, from Dr. Noah Volkow, which looked at change in brain metabolism, specifically glucose metabolism, for people exposed to cell phone radiation. It is a subtle effect, and nobody can possibly figure out what health effects might be from change in blood glucose metabolism. And yet, from the biology side, whether cancer data are classified as equivocal or beyond the range of doubt, it is this short-term, immediate, and subtle effects. That's where we really need to push the next generation of biological research on this topic. And that's where we think NTP is in a unique position to finally break through the painful, some people say horrible, debate of the last several decades of thermal versus non-thermal effects. Sort of getting away from that, getting away from temperature change, and looking at fine points. Calcium metabolism changes in the calcium channel transmission, blood glucose metabolism change, the kind of things which, while not answering the question about do cell phones cause cancer, I sure hope they don't, because everybody in my family uses them, but more importantly, what are the immediate effects and how we can take information about that and give this biological information to our friends, the engineers, so that they can divide ever better and hopefully safer devices that we all will go ahead and buy. And I will pause here and I will have more comments about biology in next panel. Thank you for your time.
Thank you. I failed to ask if the committee has any questions for either Ms. Garado or Ms. Naidenko. You could raise them now. Any questions? Oh, okay. Thank you. Our next speaker will be Dr. Deborah Davis.
Good afternoon. Nice to see so many old friends and new friends here today. I'm going to briefly talk to you about the exposure system, and tomorrow we'll be addressing some more of the biology. The controls are, for advancing the slides, are on my keyboard here.
There you go.
So I want to say this is a very important positive study that was well executed under difficult conditions. And while it is a real credit to the institution that you managed to complete it, since, as many of you know, it was first nominated for study in 1999, I think it's important to recognize, as the other speakers have indicated here, that the exposure system is, of course, not able to reflect what current exposures are. ITIS has done a masterful job in terms of the state-of-the-art system with controlled exposures that have been validated, and I salute you for doing that. At the same time, I think because of that, it's very important to recognize that historical controls are not relevant in the study. The only controls that are really relevant for comparison purposes are those in this particular study. And I was shocked to see in the draft historical controls referenced repeatedly when, as you all know, because of the unique and I would say superb engineering that went into designing these chambers, I think it would be a grave mistake and misjustice to this study to bring in historical controls for comparison. On page 29 of the current draft, it is indicated that you, of course, use the 0.08 watts per kilogram over the whole body with 1.6 watts per kilogram for 6 minutes. 6 minutes. Now, I would like to point out that you're accepting, of course, the 20 watts per kilogram averaged over 10 grams for the ears, the hands, the wrists, the feet, and the ankles. Now, I don't know how many of you have a 10-gram ear, but I don't think there are too many children out there today with that. As a consequence, I think we have to be particularly careful in looking at these data because it's a cube that the average volume is. And again, the ear is not a 10-gram cube, and very few parts of the body, including perhaps the eye, again, of a child, is a cube. I think that it's important to take note of the fact that the Ramazzini Institute has just published a peer-reviewed study which is online. Mr. Elder told me it had been pooled. I think that was a mistake in the computer system, and is online, showing that base station antennas, which can be found on rooftops and lamp poles, can exceed FCC guidelines, and the Ramazzini study is a study of base station antennas. And I'm going to show that to you very briefly. But as was just indicated, the study that you have just seen here is not designed to take into account the multiple exposures that are taking place every day, for example, in this room, or to any of you who travel in public transport, as we all do. Now, the occupational guidelines, such as they are, have never been enforced or monitored. OSHA right now, I need not tell many of you, has a skeleton of what it ever was as an agency. It has no capacity to do monitoring except for acute accidents and injuries. And the FCC website currently says that this occupational guide was later ruled to be only advisory and not mandatory. So I think it's important to recognize that the study done here, which is for public exposures, really cannot clarify what is happening in the occupational workforce, where levels right now are permitted to be much, much higher than those that have been studied here. And again, we know that this study is not going to be relevant to 5G, which in the omnibus bill that is just been passed and awaits the president's signature. That omnibus bill includes a provision to roll out 5G throughout the country, which will mean thousands upon thousands of antennas that can be placed on 30-foot tall poles, which, as you've just seen from the prior presentation, can be located right outside your balcony or your bedroom window. Finally, I'd like to comment on the value of the whole body approach that you've taken here. It is the right approach, and I think it's even more right than you might imagine. And that is because the French have a national frequency agency. This is a federal agency that is mandated to actually test phones, real phones, unlike what we do in the United States, where a single phone is given for testing. And the French system, which has now been identified as PhoneGate, has shown that when phones are tested next to the body, as opposed to in the current system that is permitted with a holster against the body, that with a separation distance, when phones are tested next to the body, they exceed the current standards. Here's what the Ramazzini results are. This is the title page from those results of base station radiation, and here is a summary of the table from those results that I will talk to you more about tomorrow, simply to indicate it's online. Finally, this is a comparison of the SAR levels that you see here from what was done in Ramazzini study, which is barely visible here, to the NTP study, to the FCC public limits, to ICNIRP, and to the FCC occupational limits that allow to the ear, again, a 10-gram volume on this beautiful baby granddaughter of mine, that her ear would be allowed to have this much exposure. You will notice, of course, the ear is quite proximate to the brain with a thinner skull. So I bring this to your attention because I think we need to address why we are not using this system, also developed by ITIS, which gives a much more precise and accurate measurement of exposures throughout the body, and I encourage further exploration of this in your future work. Thank you.
Thank you. Questions?
Yes. Yes. Niels.
Yeah, I'm not sure why you claim that this study does not cover multiple exposures. You're aware that the whole body was 6 and 10, and the spatial peak was for the rats in the range of 40 to 50, and the mice between 50 and 70 watts per kilogram. So much higher what you indicate that. That's what the study was designed for, to go as high as feasible. And these values are much higher than anybody is exposed, even occupational people are exposed to.
Absolutely, but here's my point. Current smartphones, as you know, can have 4 different antennas operating at 4 different frequencies at the same time. And as a consequence, their simultaneous operation, while it may not give you higher exposure in terms of watts, it cannot, as we know, it cannot exceed the thermal levels. We have evidence from other studies that have been done by Tillman, by Lerkel, and most recently the Ramazzini Institute, showing that there is a synergy when you combine EMF with RF or chemicals with RF or EMF. And it's this synergy that we cannot evaluate in a study like this. It's not possible.
So you're not talking about multiple exposures from different RF sources, you talk from combination of different agent with RF?
Both, actually. Different agents for sure. We're talking about ENU in the case of Tillman, EMU, and RF in the case of Ramazzini. We're talking about the study that was done with Safiati, looking at exposure to ionizing radiation and non-ionizing. So as you know very well, the real world is complicated. The elegance of the studies that we do here, which you can't compete with, cannot approximate what the real world is. And I would remind all of you that while this study was first discussed at the Senate hearings where Dr. Bucher and I appeared with Dr. Olderenko in 2009, in the meantime, the rate of use of this technology has exploded, so that since 2007, it has doubled more than 3 times. So the rate of increase is just huge, and we do not have the capacity within our experimental models to fully approximate what the human exposures are going to be. And I would, with all due respect, disagree that it's great that we have all these exposures right now. I think we need to ask, what do they mean for our health for the long term? And in particular, what could they mean to our children who are growing up in a sea of radio frequency radiation that has never been tested. This Alexa that you saw there being sung to by this little child, that is supposed to be used 20 centimeters from an adult male SAM body, the big guy. And children playing with these things, having them close to their bodies, or children in public schools now being given iPads in kindergarten and preschool so that you will help compensate for their deficient backgrounds. I think we have a problem in what's going on with these exposures, and I really think it's important to acknowledge that the real world today is so much more complex than we can approximate in these studies.
Thank you. You've got some time tomorrow too, Dr. Davis. So.
Thank you, Dr.
Thank you. Thank you. Okay, we'll move on. Kevin Motus.
So hi, my name's Kevin Modis. I'm with California Brain Tumor Association, and we support individuals who have developed brain tumors from cell phone radiation. So we see the devastation that occurs on an individual, a family, and a community level. So we came here to thank you, the National Toxicology Program, for embarking on this study and following it through so conscientiously. And for designing a study that could show really what we're seeing out there, and showing importantly the DNA damage that is at the basis of the carcinogenic effect, to seeing effects to the cells that we know in humans represents the myelin, and we're seeing neurological effects with folks as well. And understand this, we don't just work with brain tumors, we work with people who are developing microwave sickness or electrosensitivity or electrohypersensitivity, however you want to label it. A day doesn't go by when we're not contacted by someone who's become very sick from wireless, and usually it's a young person in their prime, and their lives have been ruined. They've developed, fatigue and headaches and nausea, dizziness, heart palpitations, tingling in their skin, mental confusion, and it's ruined their life. They lose everything. They lose their job. They lose oftentimes their family, their relationship. They have to move out to rural areas to get some kind of respite. So the reality is, the human evidence is there. We've been going about while this study has been being done, embarking on a human study, and we know what the results are. We know people are getting sick, and we're very grateful for this study to show the biological basis for this. We're also grateful for this study because it shows that the cells that became cancerous in this study, align with the type of study of cells in human studies that became cancerous. And now with the Ramazzini study, another study showing the same cells being affected. So now we have this biological confirmation to the human studies we've had for years, and in alignment with what the human experience that we're seeing out there, like really in the field. So that we have enough to really move to reclassify wireless as a class 1 human carcinogen that should be warned about and should be minimized. And so I'm really here to say thank you. Now, it's hard for us to hear the FDA criticize this study and your techniques, because the techniques seem impeccable, based on the fact that, we're not seeing an increase in brain tumors overall, so we can dismiss all of this. It's a mute discussion. But I would beg to differ that there is a clear increase in brain tumors in the 3 areas of the brain that get the most cell phone use. That is the frontal lobe, the cerebellum, the temporal lobe. We have one study showing an increase of between 20% and 400% in the 10 years before 2006. Now we know there's been an explosion in use since 2006, so you can imagine the continuing increase. And people can relate to that because people are seeing people get brain tumors now that used to be very rare, along with tumors in the in the head, behind the eye and in the neck. So I really want to put that out there. We have another study also that's showing that our heavy users, that is our children, unfortunately, that brain cancer is now the #1 cancer for children 15 to 19. And it wasn't that way when we first introduced wireless. And that brain cancer is now one of the top cancers, the top 3 cancers, all the way up to age 39. So we believe we do have an epidemic going on, and we believe the industry is pointing to the overall rates for the whole head and staying away from the 3 areas that get the most cell phone radiation. We also don't appreciate the FDA saying that, well, this study isn't relevant because it's looking at 9 hours of use, and no one uses 9 hours. Well, I have to tell you, we work with salesmen and lawyers and, unfortunately, children that are using 9 hours a day. But we know it wasn't about the 9 hours a day, and this study was designed really ingeniously to look at whole body exposures, and that's really what we're dealing with today. And the new introduction of products is really focused on these continual long-term exposures, Wi-Fi, and now cell towers with 5G. And we are very concerned and so glad the NTP study comes out, because we think it's very relevant, because NTP showed a dose response, and that is the more wireless you have, the more chances of getting cancerous effects. And by layering these different types of wireless, low frequencies that we're still going to have with 3G and 4G transmitters, with 5G high frequency, high intensity transmitters on top of it, we're likely to push people closer to disease even faster. And we're glad the NTP has put that out there as an important factor. And my previous speakers have talked about the other criticism with the FDA is, well, this doesn't really emulate personal use, and it's been talked about a lot, that people are exposed now to many devices. I kind of want to make that point that most people using a cell phone now are also using a Bluetooth in their ear. They have a wireless laptop in front of them. They're sitting near a wireless router. They have maybe a wireless burglar system in their house. They may have a smart meter on their home. And then with 5G, they're going to have a cell tower transmitter in front of most of their homes, emitting another type of frequency. And then with our self-driving cars, the vision is 8 radar per car and sensors inside of the cars. So you're talking about a really thorough microwaving of our population with low frequencies, high frequencies. And I would say this study doesn't take into account multiple types of frequencies at multiple types of intensities hitting different parts of the bodies and how that affects the body. And I think, frankly, it's irresponsible that the FCC on Thursday passed the wireless infrastructure streamlining order, which is basically going to say legally we cannot take into account this study and these findings, that we're going to roll this out exempt from any kind of environmental review. I think, there's been talk about reaching out to the FCC, but the FCC is hiding health effects and has been hiding health effects, in my opinion, and passing an order like this kind of reflects that. I'm also worried about 4986 was passed on Thursday as well. That's the congressional bill that's going to say, yes, again, we're going to streamline this, we're going to make it exempt from environmental review, and we're just going to see what happens with the American people. I think this is truly reckless and irresponsible. And I know that this program came out with the findings earlier because they were very concerned, you guys were very concerned about brain tumors and cell phones and brain tumors. I ask that you re-engage and recommit yourself to taking a stand here, because we know the FCC is an industry-compromised agency, that the chairman's the former Verizon attorney, and the one that rolled out 5G was the former president of CTIA, the wireless lobby group. We don't have really anyone fighting for the American people right now who are getting sick from this and who we deal with every day. And understand this, I have formal testimonies from people from every state in the union. And this really needs to be addressed, and we really want you to take a stand. But the story of wireless is really the story about industry infiltration to our research programs, interference with our research efforts, the choking off of research funding dollars to not look at this. It's simply unacceptable, and as part of putting out the findings, we also want you to say, this is a big part of it.
Mr. Modis, I'm going to interrupt you now. We're several minutes past your time, but you're on our schedule tomorrow as well, so I thank you. Does anybody have any questions for him right now? All right, well, thank you.
I just want one closing statement. I just hope this program tries to insulate itself from industry influence, and I was hoping that I would see even more public university scientists and doctors on the panels, in addition to all the private industry scientists that I'm seeing on both of the panels. So, thank you.
Okay, next will be Paul Hero from McGill University.
Good afternoon. I was trained as a physicist, and I teach graduate courses in toxicology and health effects of electromagnetism at the Faculty of Medicine, Montreal, Canada. The NTP reverberation chamber delivered to the test animals a stable challenge over a specific integrated time frame, related to the necessity for reverberation room mixing. This is important because various physiological systems in the animals react to the EMF challenge over times that range from nanoseconds to months. A variation of plus minus 2.5 decibels in the exposure was quoted in the report, depending mainly on the location in the cage racks, and I assume that this could have been reduced by using larger chambers, expensive, so that the objects would occupy less of the total chamber volume. IEC 61421 now has a requirement for less than 8% occupation. Nevertheless, this plus or minus 2.5 decibel is excellent performance in terms of heat and generic modulation. So it's an incredible success. But although the study was very well conducted, it does show its age philosophically by its overemphasis on heat. The survival is higher in the non-exposed animals in the NTP results, and this is commonly considered in the media to be an artifact. I submit that it's not. I know of 2 other large animal studies that show indications of the same tendency, and this lifespan lengthening is compatible with interference of EMR, with oxidative phosphorylation and activation of physiological defenses. Lebars published in 1983 a 20-month study in which 220 AKR female mice, which spontaneously developed leukemia, exposed to 50 Hz electric fields of 50 kV per meter, rather large field. The fields were applied by large diameter tubular electrodes a considerable distance apart, which meant field uniformity characteristics similar or better than those of the NTP test cells. The animals were 4 groups of 55 mice, 2 sham, 1 exposed 8 hours per day, and 1 18 hours per day. For both the exposed groups, mortality appeared 1 month later than in the sham groups. These mice live a year approximately, which showed the survival expected in this species. At 7 months, the number of dead animals in the sham group, 10, is significantly higher than the exposed 2. In June 1992, exposure administered in a circularly polarized waveguide operating at 2.4 gigahertz and delivering 0.5 watts per kilo to 0.4 watts per kilo, 24.5 hours a day for 25 months. The system delivered exposures less reliably than the NTP setup because they relied on a sum of measurements rather than direct dipole measurements. Still, there is some indication that the survival times were longer in the I'm quoting the study here, and median survival time was 688 days for exposed animals and 663 days for the sham exposed. The NTP rat and mouse data both show a trend towards longer survivals with increasing exposures. The trends vary in strength but are valid as a function of intensity for all rats, all mice, all GSM, all CDMA, all male, and all female. So increased exposure survival occurs in 3 different studies where a number of rats or mice were respectable. Lebars, 2 separate experiments involving a total of 220 female AKR mice. Chu, 200 male rats. NTP, 421 CDMA mice, 420 GSM mice, 420 CDMA rats, and 420 GSM rats. That's a lot of rats and mice. A second interesting aspect is that the effects are stronger for males and females. In rat studies, either with GSM or CDMA, exposed males reap a longer survival advantage than females from the exposure. In both mice studies, either with GSM or CDMA, exposed males also show a slightly larger survival advantage than females from the exposure. This sex difference is expected because males rely on a higher metabolic rate. Therefore, males would be more affected by the fields. This suggests that the effect is not an artifact and that more precise exposure systems allow the detection of the activation of basic cellular defenses, which we documented in vitro in our experiments in our lab. In a sense, you know, this survival issue being treated as a problem is that the NTP is being criticized for its success. I note in the reports there is no mention of control of background ELF environment. This, I haven't seen any measurements of this. ELF components that dominate the biological impacts of these systems are very important. So the exposure system was very successful in displaying aspects of the EMF problem with a very specific challenge. We see that there's a difference in the effect of GSM and CDMA, and so the details of the exposure become very, very significant and important. So I don't know whether the US has enough money using animal studies to increase the focus and the precision of our knowledge in this area. And I'm right on time. Thank you.
Thank you very much. Questions from the panel? Okay.
I just comment that there is additional data of other kinds that also shows a positive effect in terms of reduced incidence of problems with some exposure as opposed to heavy exposure. So that's something to be discussed another time.
Okay, so we are going to move on to the next item on the agenda, which is to hear from our peer reviewers. We've asked for each of the peer reviewers to provide their comments in an order of about 10 minutes each. And Dr. Barnes, we'll start with you.
All right. Well, first, I think the one is this study was very well done in terms of doing what it set out to do. There can be a separate set of questions of whether the problem was defined the way you would define it today, and I think that's not so, but that's to be expected if this thing was started in 1999 or in 2006. So first, I think, yes, if you take SAR as the critical parameter to define, they've done a very good job of seeing both what the exposure distributions were, that the average values were what they stated. It's been well tested and well monitored throughout the study. So I think it has done what they set out to do extremely well, and I don't imagine a way of doing it better. At the same time, if one were to design the study today, I can think of a variety of additional experiments that I would, and parameters that I'd think one want to control. And just mentioned with the fact that there were measurements not stated, at least made on ELF, and we've seen stuff in my lab of changing static magnetic field, and its orientation makes a difference. And this certainly was not in vogue at the time that this study was put together. But that sort of thing, and things dealing with reactive oxygen and other translations from physics to chemistry, chemistry to biology, so forth, are things that one might well build into a study that's going forward. Similarly, looking in more detail at the problems of feed-forward and feedback loops with regard to correction and time dependence of what's going on is something which, for subtle effects, could be looked at in much more detail at another stage. But overall, I think that it would be complemented on a very thoroughly done study that carried out, to at least the best I can tell, what you set out to do.
Thank you. Any questions for Dr. Barnes? Dr. Booker.
Thank you. Could you elaborate a little bit on what you just mentioned at the end, the feed-forward aspects of this? Because we're very much interested in improved studies.
Well, we have feed-forward loops, for example. You see something, you jump later. You wind up, you eat something, your stomach sends signals forward that says to prepare for that. And we certainly have elaborate communication systems inside the body. So, and maybe the classic example of that is the acupuncture points, where you stimulate a nerve at one place and it causes effects at other parts of the body. So there is certainly, I think, one of the more exciting things that we have is the opportunity to use electric and magnetic signals as a way of converting those signals to biological signals in the chemical realm that the body already knows how to use. And I think that's an exciting possibility. Examples of this would be optimizing, say, suppression of cancer growth rate or suppression of an overactive immune system. At the other time, we see that we can stimulate growth faster. It's done in the bone studies. All right, I think there is a whole lot to do in terms of looking at how you translate either an electric or a magnetic signal into a chemical signal to do interesting things. And I just make one comment along the line. In my lab, we've shown that we can both inhibit and accelerate the growth of fibrosarcoma cells by simply changing the static magnetic field. And if you'd asked me about that 5 years ago, I'd told you it was nonsense. You wouldn't think that anything like this would happen. But we're seeing it in a systematic way. So I think there's a lot of potential things to do that are very interesting.
Thank you. Any other questions for Dr. Barnes? Okay, we will move on. Dr. Lin.
Okay, thank you. Well, I'd like to applaud the NIEHS and NTP for having sponsored the research and conducted the cell phone radiation studies. It is important for the U.S. government to step in to conduct such a research program and not leave the matter entirely to industry. Appreciate the completion of this study will not signify the end of U.S. government's engagement in or role in supporting RF biological responses research, because we are all being exposed to increasingly more and more RF radiation every day. It is well known that the NTP cell phone RF studies is by far the largest study of its kind. It was rather expensive and took a long time to get to this point. There may even be better ways to do it. Nevertheless, the study points out to the fact that prolonged exposure to RF radiation at levels that are about 3 times above current RF exposure guidelines or rules may lead to tumor development in particular, schwannomas in the heart tissues of rats, and to some degree, gliomas in the brain. Our charge in this part of the panel is to assess the reverberation chamber, which in short RC, methods and technology for evaluating the carcinogenic effects of cell phone RF radiation exposure on rats and mice. It appears that the RF was selected a priori for the project. It is not clear. It is not clear RC is the optimal technology for the project, or whether other competing technologies such as circular waveguides or small rectangular multimodal chambers were seriously considered for exposing freely moving animals inside their own cages. Descriptions in the reports of what was implemented are fairly clear. Measurement techniques are accurate to the extent that they were applied. However, there are limitations. The large number of RCs constructed specifically for this project are the most expensive and one-time use or single-purpose equipment or facility for bioeffect research. They likely would not be used for another project. Thus, they would end up as being, in quotes, white elephants by default. If they were not, in fact, they may not already been scrapped. It appears that NIEHS NTP has moved on to other types of exposure chambers to continue its RF research. It is generally acknowledged that the RCs could provide substantially uniform average field distributions in the absence of any test object, a feature that works well for testing electromagnetic compatibility, immunity, and interference of electromagnetic devices or systems under test. In those cases, RF energy absorption by the device or system is designed to be low, energy absorption by the devices or systems generally are by scattered fields inside the RC. In contrast, red mouse bodies are major RF energy absorbers in RCs, a very different interacting mechanism of fields inside RCs. The presence of free roaming animals made inside the cage would render the exposure field substantially less uniform compared to empty RCs. Thus, while much time and technical support and efforts were spent on achieving RF field homogeneity and so-called isotropy of RCs, it is not obvious whether or not the RC approach had any advantage over simpler approaches, especially since average or whole body specific absorption of SARs is used as a metric for exposure. Also, it is unfortunate that the reports chose to mix dB and linear percentage scales to describe RF fields and exposures, which makes them not as easy to discern or compare when reading it if it is not entirely confusing. It would be more appropriate to describe uniformity using average field distributions than homogeneity and isotropy. It is not clear how isotropy, meaning independence of orientation inside the RC, was determined for each animal. Moreover, field distortions produced by the choked and shielded automatic watering system when the RCs were fully loaded with animals do not appear to have been quantified. Thus, their impact on field uniformity require further assessment or clarification. The key exposure parameters chosen for the dosimetry quantity is the, as we know, average or whole body SAR. The choice of the maximum exposure level was made to prevent excessive, meaning not greater than 1 degree Celsius rodent body temperature. This was guided, as we have heard, by RF-induced temperature rises from homogeneous phantom measurements made during pilot study, which used liquid-filled plastic bottles, not freely moving animals. Actually, one can raise a question as to why were not body-shaped plastic models chosen for this purpose. Measured exposure field levels had 100% variations in empty RCs. You can see this in figure 2 or table 3. It was not measured with the watering system in place, which likely would have contributed additional field variations to scattering by the metallic shielding of the watering system. It is noteworthy that RF power deposition occurs instantaneously, while heat transfer and temperature change occur over time and take considerable longer length of time. The reports indicated that the RF field uniformity of 0.46 dB, or by my calculation 11%, or 0.4 dB, or 10% for rats and mice respectively, were achieved throughout the RC volume using liquid-filled round plastic bottles. Clearly, round bottles do not provide realistic simulation of body's shape or configuration as far as SAR variation is concerned, let alone the SAR distribution inside the animal. In fact, the calculated peak spatial SAR may be 6 to 11 times higher than whole body SAR, depending on body mass and tissue location. Again, this can be seen in the Gong publication. A similar situation was revealed for organ-specific SARs. Moreover, posture variations has been shown to introduce large changes to both whole body SAR and peak spatial SAR. Note that a rat's body resonates at about 900 megahertz. The whole body SAR may vary by as much as 5-fold in this case. Again, one can see this in figure 4 of Gong's paper. The resonant absorption scenario did not arise for mice, which may be one reason why observed biological responses were different for mice. It is not obvious that this five-fold difference in absorption was compensated for by adjusting field levels between 203 to 315 volts per meter, because if one simply square these, we get number 4 or number 9, leave out the zeros, which is only a little bit more than 2.5 times, not anywhere near 5 times. The situation is confounded by the differentials between peak spatial SAR and whole body SAR. Thus, given the observed tissue site dependent biological findings, an analysis examining any correlation between peak spatial SAR and or organ SAR and biological responses should have been included in the project report. What influences, if any, the differential SARs may have on observed tumor incidences? The methodologies and paradigm and protocols used are reasonable but equivocal. Aside from GSM and CDMA modulations, the exposure regime included 10 minutes on and 10 minutes off for 18 hours per day. It is not clear if the research study was intended for cell phone or base station RF exposures, whether it was for near field or far field exposure scenarios. Temporal and spatial averaging was relied upon to maintain targeted whole body temperature level, as well as SAR, based on, again, on bottle phantoms during the entire animal's life. However, this approach ignores anatomy-related responses of the animal to RF SAR and its distribution inside the animal, or as a function of time or age, especially if one considers the potential ongoing thermal regulation of the exposed rodents. It appears that the experimental protocol did not include physiological monitoring or visual observation of animal behavior at any time during the 2-year study. For example, did animals exhibit any preference for cage location or area or posture or activity during those periods? It would have been confirmative to have made quick periodic measurements of the, say, tympanic temperature throughout the study as a monitor for core temperature. It appears that the continuous or ongoing monitoring was not part of the protocol. Apparently, heart rate was not monitored to help assess any stress or thermal regulatory responses in these exposed animals. As I had asked previously, it wasn't clear from the reports whether the environmental monitoring protocol included the mechanical disturbances such as noise in the control and exposed RCs. And it is not obvious from the reports whether molsters were in operation both in control and exposure RCs. And also, I think it is important to have measured the spectrum, the frequency of the noise that was in these chambers. Now, whether the finding of schwannomas in male, well, let me say it again. While the finding of schwannomas in male, not female rats, may be explained in terms of gender differences in response, the null findings in both male and female mice, even though they were exposed to higher SARs than the rats, are intriguing. Aside from specific differences or species differences in the animals, it is possible that SARs may not be what was reported. It is perplexing to see reports of SAR dependence of schwannomas in rats. Since this project involved whole body SAR or whole body exposure to RF energy, in addition to cancer from specific sites, a statistical analysis of total primary tumors observed in tissues and organs should be included in the report for exposed versus sham exposed animals. Also, given the observed tissue site dependence of tumor development, in particular the schwannomas as well as the brain gliomas, an analysis examining any correlation between peak or organ-specific SAR and tumor incidence should have been included in project reports. It is important to assess what influence they might have had, especially if we consider these localized absorptions. Finally, I'd like to note that the current RF rules or guidelines are 1.6 or 2.0 watts per kilogram are promulgated with a reduction of safety of 50 or reduction factor of 50 as a safety margin. I think the findings that we have been seeing during at least this day's presentations, that exposure levels at only 3 times above current guidelines are significant and should be important for all of us who are concerned about potential effects of RF exposure on humans. Thank you.
Thank you. We will, after the next presentation, we'll then have a discussion where some of these questions can be addressed. And Dr. Booker, you want to respond to any specific points here?
So, thank you for your comments. One of the things that we've been working on and we wanted to get a good handle on at this meeting was how we could improve the report with respect to communicating the information that you talked about and communicating the information that Miles and others have talked about, and John Ladbury earlier. It sounded to me like when you were going through your comments that many of the things that you had picked up on were in fact included in some of the talks and some of the had been addressed, and I recognize that they have not been in the report currently. We would very much like suggestions specifically as to how we could encapsulate more of this information, specifically with respect to the peak spatial SARs. I'm not, we have the organ-specific data in one of the manuscripts. I don't know exactly how much we can get into the extension beyond measurements that have already been modeled, but clearly that seems like that's going to be an important thing in relation to the rest of the communication of these findings as how people would actually use them in a public health context. But I'd be happy to have reactions from Niels or Miles if I'm misinterpreting what I think I just heard.
I think we have the data, and Miles have shown of the spatial peak. So these data are available. Is it now time to discuss some of his points, or do you want to?
Well, I would suggest that we wait until after we've heard from our last reviewer, then we can come back to some of these issues, especially recurring issues that are brought up, and then we can get into that. Okay, so Dr. Chiorti.
So yeah, on my end, I would like to congratulate you on a very thorough study. No major comments at all regarding the technical part of the study. My comments are only related to the report. So these are comments I put together before I came here when I read the papers and the report. And a lot of these have been answered through your presentations. Just a general comment before I start going through my comments, that the question is, how do we catch up with technology in general? I mean, the study delivered 100% of what you promised in doing. Right now, 2G is not even used. At the university, I'm from the world of academia, we're trying to do research that we want to think it's 10 years ahead. So like in my group, we're developing wearables and implants with the hope that 10 years from now, people are going to use it. So these dosimetry studies, how can they catch up with technology? This is just a general comment. So back to my specific comments regarding the report. Dr. Lin had several questions today about the exposure system and the cycles. I agree with that. It is not very clear in the report, especially in the abstract. It says that GSM and CDMA signals are cycled every 10 minutes. So just by reading the abstract, you're certain that you're just switching from GSM to CDMA, which I know it's not true. I read the papers, and in the papers it's very clear. Another question I had was about the RF sensors. You're using E-field and H-field sensors to make sure that everything is in place. I understand that the exact location doesn't matter because everything is supposed to be homogeneous in the reverberation chamber, but still I would like to see some details about where exactly they were placed and why did you select the locations as such. Regarding the environmental conditions, I know you had several sensors trying to maintain them at a certain level. My question is, how did you decide on these specific environmental conditions? And if you had decided them to be somehow different, would you expect something to be different in the results? One more point relates to the size of the cage. So one point mentioned in the report is that you didn't use restrained animals because that would increase the stress levels. So I understand that animals are free-moving, so stress levels are reduced, but still they are within a cage for 2 years. So I was wondering whether stress in this case is somehow a confounder. One other question relates to the antenna design. So I'm an antenna person. I understand that the point is having a homogeneous field within the reverberation chamber at the end of the day. But even though antenna design and antenna location is within a bullet point list that you have in the report saying that antenna design and location is important, there is no information as to why or what was the antenna that was selected. I saw the antenna today, but I hadn't seen it in any of the papers or the reports, or at least what's the pattern, the radiation pattern of the antenna. One more point. So you're mentioning about 2.5 dB of variation in the fields, and that's why you're rotating the cages. So I was wondering, how was the routine selected for this rotation? 10-minute cycling, you already answered the question, but it was like natural to think like, why 10 minutes, why not 8 or 12? But this has been mentioned already. One question that I had, it's not really critical, but I saw that NIEHS carried out studies using water bottles as phantoms, but then at ITIS, you actually use the phantoms, the tissue emulating materials. So I was wondering, how did the two studies compare from a phantom point of view? So water bottles versus phantoms that emulate the tissues properly. Yeah, the rest is mostly editorial comments, which I have included in my report. So yeah, I don't think it's worth mentioning them now. You can go through the report. Right. Thank you.
Well, thank you. So this is now an opportunity for all of our panelists to ask questions of the other, especially panel 2 to ask questions of panel 1, as well as the NIEHS staff and consultants. I would first like to congratulate everybody for really outstanding presentations today of a great deal of information. It's a little hard as a biologist to get my head around all of this, and yet as a biologist, first and foremost, is exposure and dose, and so I think that this is very helpful to hear in such detail how these exposures were done. I'm going to open up. I have just one general question about. It's been raised a number of times that the design of the experiments was perfectly appropriate for the time and for the technology available at the time, but we've also heard that the technologies have changed and are changing. And the question that I have as a toxicologist for those that are looking at exposures, are those changes in the nature of RF dosimetry with new technologies, are they in a way that would suggest that the previous technologies were overestimating health effects? Are the nature of the exposure is likely to be better now or worse now? And given what little we know, frankly, about the biological mechanisms of this. So the question are the differences that we would have today for exposures quantitatively different or and/or qualitatively different from the types of exposures that were done at the time. And I'll just throw that out to the experts because I have not a clue. Dr. Barnes.
Well, I think one of the things is that we do have variations in how the power is distributed and deposited as a function of frequency. So if you look at 900, 800, and the new coming 5G or millimeter wave, you get a different distribution. From a biological point of view, you have different sensors in different places in the body. You have different time constants for different responses. And there's some indication that there are resonances that need to be dealt with so that things can be frequency specific. So the kind of detail that describes how you go from the physics through the chemistry to the biology can change with that. Now, on the other side of it, we're seeing some of the same things with static magnetic fields that my colleagues have seen at the RF that are being seen in the terahertz region and have been seen in the infrared. So there may very well be common responses across a lot of this. In particular, I'm talking about changes in reactive oxygen, membrane potentials, calcium efflux, etc. So there are some commonalities, but I don't think we understand how you go from the physics to what is happening in the biochemistry well enough yet to say which of these things are frequency specific, which of these things are amplitude dependent, how they're time dependent. Yet I think that's part of the biology we don't know.
I don't know which question or which topic I should address first. Maybe the easiest one for me would be the question regarding bottles versus more anatomical phantoms in the experimental setup. What we didn't mention in the report is that we carefully evaluated if there's any coupling between the phantoms. And by purpose, the cages were separated in such a way that we can exclude coupling. And when we can exclude coupling, then it doesn't matter how the energy is scattered. It's only a question of how much energy is absorbed. And that's why the bottle phantoms were optimized to absorb the same amount of energy than a rat. And that the bottles were only used for validating the numerical dosimetry. The main body of dosimetric data is coming from the modeling. Bottles were only used for the validation, were not flowing into the dosimetry.
Well, it may be because in one way, I may consider myself as a Johnny come lately as far as the project is concerned. But the published dosimetry was dated at 2017. This work was started 10 years before that with the bottles. So which one came first? How did you validate the dosimetry with the bottle when on paper there was no dosimetry done at that point?
Yeah, that has the effect that Myers prefers to build instead of writing. So that's why the papers became very, very late. So when we did the evaluation beginning of 2000, then we already did dosimetric data, not with all the posters, etc., because technology wasn't available, but we did the animal dosimetry. And from the animal dosimetry, the bottle were derived.
Okay, let's get to the more technical aspect of it. As the dosimetry eventually indicated, the posture, anatomy, or the geometry of the animal actually makes a difference for dosimetry. Therefore, a round bottle is not the animal-shaped body. So clearly there's a difference because your own data suggests they are not the same.
But remember, so the bottles were once used to validate that the exposure was uniform throughout the chamber. Not affected by the presence of the different animals.
Surely, of course they are.
No,
the presence of the animals, that's why I indicated by all the hearing I have had, all the readings I have read, after the watering system, for example, I use as an example, has been installed, there was no indication there was additional field measurements made. At least no one has mentioned that that was done.
But the field measurements were done at the end, before the exposure started. And NIST, even at the end, they have redone the entire field measurements when the system was.
Now, that was with everything installed in place?
Everything installed, the cages, the food, the water system, and the animals were replaced by the bottles, but I tried to explain why the bottles were an acceptable simulation of the living animal in the system.
No. On the empty chamber field, that's why with the all the installation that was done inside it, you can certainly measure the external fields. But the field inside the animal is posture and geometry dependent. So that part of it, I think, was not quite cared for or was clearly described, at least by what I cannot guess what has been done, but in terms of what has been said, that's not the case.
Yeah, so we take the point that we need to describe that better.
Mark Klein, Wake Forest. So as somebody who works in ionizing radiation, I just wanted to mention that dosimetry is being used incorrectly in some of the talk around the table. In the report, it's used a little more carefully, but dosimetry is the measurement of the dose. And so modeling, a mathematical model of what you think the dose is not dosimetry, it's modeling. So I just want to throw that out there.
That I have to disagree. As you indicated, you come from the ionizing radiation domain. Non-ionizing radiation do not behave the same way. In other words, in the ionizing radiation case, you have almost straight line propagation of the radiation field. In the non-ionizing, in this case, the field changes directions as it encounters various objects. So those are totally different. So what the terminology, those might be used in one case. In fact, the units can be used the same way. In ionizing radiation, watts per kilogram is also used. Okay, temperature rise-dependent measurement is also used in ionizing radiation, but they mean entirely different things. I'm sorry, I have to contradict that point.
Okay, I'm gonna stick to my guns. The dosimetry is the measurement of the dose.
Yes, measurement of dose. Then it comes to a definition. What do you mean by the dose? In this case, the two doses are different. One can also bring in chemical or pharmaceutical dose. Those are entirely different. Again, the word dose is used as well in those cases. While I have the microphone on, could I say a little bit about the generation of technology, whether the NTP study is appropriate for what it was intended for? Clearly, we all recognize the fact that, especially in the wireless communication domain, there are changes not every day, at least almost every month. So we are now in this situation whereby the 5G is on the verge of being rolled out. So the technology that is perhaps most relevant is the 3G technology. In many cases, I wouldn't be surprised some of us still have that technology in our pocket. So this is still current. The 2G is by and large no longer, except in some isolated situations around the world. So when you design a study, you want to find out, in this particular case, as to what happens if you expose the whole body of the animal to that technology over a period of time. Does cancer develop or not? Or under what circumstances? I think that answers a very, very important question that does not answer the question of today, but that answers a question that we've been faced with. A lot of people, we're talking about the the number of users had tripled. In fact, in the United States, there are more cell phones today than population. But 10 years ago, that wasn't the case. But nevertheless, there was a substantial portion of the population that was already exposed at that time. So I think, as much as I can discern, that was the impetus to carry out the study to see what might be happening with this technology. So from that perspective, I would not have any hesitance as to the validity of having conducted such a study.
Sorry. Questions from panel two panel one members, anybody? No? Jack.
So I have a question. It was mainly in the design. So even going way back, and this is kind of an odd question because we have a long history of 2-year bioassays here. But I'm sure at the time of 2007, you were questioning whether or not to use 2-year bioassays. And I just wondered if the design of this study was influenced or hindered in some way by constraining it to this 2-year bioassay. And maybe you see what I'm saying? No. I'm just, because a lot of the historical control data was that you wanted to do it exactly the same way, or were there, I guess the better question is, were there any constraints that you would have, to the study, that it would have been, or in the future, designed in a different way to do it? I give you one example, is everything's after 2 years, okay? There's a lot of carcinogenicity studies that have shown tumor development, greater number of tumors after a 2-year period of time. So.
I would just comment on that in an area where I think you were somewhat clairvoyant in the design of the study, because when this was designed, starting studies in utero was almost unheard of. And we now recognize that the period of vulnerability in development is early life exposures, especially for many types of cancer. So I applaud the NTP for looking forward and starting this, because Jack, in those days, virtually every study that was done was, on weaned animals, starting the exposure at the time of weaning.
Yeah, I'm not criticizing that at all. There was a lot of fantastic things that was all done. I'm just asking a question in the design, and was there any constraints to that besides money and time?
Well, those are significant, money and time, and I mean, we did try to increase the animal number sizes to a level that gave us probably the best efficiency with respect to the numbers of animals versus the cost associated with the increase in power that you get going much beyond 100, because it diminishes rapidly. We also recognize that there are situations where where 2-year bioassays have been converted to lifetime bioassays, but with respect to the NTP studies we have, and Keith Shockley can talk to this point tomorrow, there's an issue with respect to the ability to carry out properly survival-adjusted statistics under the conditions in which you have no terminal point from which to gauge the proportion of lifespan to attribute each animal's risk in that overall tumor incidence calculation. So you're right, there's some tumors that will appear very late. The other problem is that you get an increased onset in spontaneous tumors the later the animals go. So sometimes you'll actually lose the ability to pick up a tumor, a chemical or a physical agent-related response. So it's certainly an area of debate, and I don't think we have the best answer.
So I think you answered it, that there weren't, and that's what I wanted to say.
Dr. Davis, you have a question for the panel?
Actually, I have a response to a question that you asked, Dr. Eaton. You asked about what data we have on the relative potency of newer technologies compared to older. And the presumption that's been driving much of the discussion has been that with the weaker power, you will get less of a response. And we held an expert forum with the Israel Institute for Advanced Study at the Hebrew University of Jerusalem approximately a year ago. And in that forum, we heard from DARPA-supported physicists in Israel at the Weizmann Institute. And they reported that they have done studies on 5G that have found just the opposite of what we would have expected, which is that the weaker power, but the higher frequency is more biologically potent. And so, contrary to what we would have thought, that 3G and 4G are going to be safer inherently because the power is weaker, which we know to be the case. The power is weaker. It may be that some other properties of the frequency make it more biologically potent. And specifically, there's a publication that has just appeared in the Journal of Environmental Research that I'll make available to the panel. It just appeared now on exactly this point, showing that 5G resonates at a 1/64th of an inch into the human skin, where the sweat ducts of the human skin function like resonant antennas. And of course, we have millions of sweat ducts. And one doesn't know what that might mean, for example, to melanocytes or other cells that maybe get promoted by that exposure. But the technology is currently being used by the Defense Department. You can find this on their own YouTube videos, because it's the basis of a weapon for crowd control at higher powers that makes skin feel like it's on fire. So all of that, it makes a more complicated story, as Dr. Barnes has said. Some signals may be beneficial, some may be harmful, but it's not the power. It may be the pulse, it may be the information content, and maybe other properties of the technology that we have to look at. And again, your job will get more complicated very fast.
Thank you for that comment. Other questions? Dr. Melnick?
Just want to add a comment, because there seems to be a lot of chatter about 2G, 3G, 5G. What would you have done differently? The objective of this study, like all toxicology studies, is to test the null hypothesis. And that's what was confronted when this study was designed in the early 2000s, because people were saying this is non-ionizing radiation, there's no possibility for an adverse biological effect, and therefore the study was designed to challenge that hypothesis. That's why it was as many hours per day, because we knew there was a limitation on how high of an exposure intensity you could go without causing a heating effect. In chemical studies, you can go higher than what is an occupational exposure limit, but here the limit is on heating. So you want to challenge the null hypothesis to the extent possible. Then, if you see an effect, what is the dose-response relationship? Because that kind of information can then be used for a quantitative risk assessment. And that's what would lead to FCC perhaps making some kind of decisions. In terms of what this means then for 5G, is that the assumption still exists. There's no reason to believe that there is an effect. That's why this study is so important. It says the null hypothesis has been disproven. Therefore, if we're going to roll out new technologies, shouldn't we know whether there are health effects before exposing the general population?
Thank you. Dr. Koester.
I want to quickly support this argument, and we are in the challenge in the way that we need to come up with exposure scenarios for the new technologies, and we are doing that. And I must admit that this would have the label 4G when we would do it today, but essentially the exposure would be very, very similar. Would have slightly different modulations to account for that, but essentially would be the same. So I think that from a scientific point of view, translation is straightforward. From a marketing or the communication point of view, that's more difficult to say that 2G is the same as 4G or 5G.
Thank you. So I want to make sure that all of the panelists have had an opportunity to ask questions of the others, that everybody is satisfied that their questions have been answered. I will bring us back to the objective here, which is a review of this report, not so much all of the things that might happen in the future, but how can we make sure that the study report, when it's in its final form, accurately reflects the data, points out the challenges and uncertainties that have been raised and in terms of the exposures. I think, from my own sense of sitting through all of this and hearing about the rather intense evaluation of all the physical aspects of the exposure chamber, the reverberation chamber, I think that there are some needs to improve some aspects of the communications about what it really has done and has not done. I heard a sense that there was fairly strong praise for the NTP program and their designers and consultants about doing pretty much about the best job you could do in constructing a very challenging exposure situation, especially for the NTP that's mostly used to giving animals chemicals. It's a little easier. So I did not hear any what I would call fatal flaws raised in terms of the exposure. Certainly there were concerns about certain aspects of it. I think many of those have been clarified. But I want to make sure before we leave today that everybody is on the panel is satisfied that they've had whatever questions they might have had answered regarding the reverberation chamber and the exposures, because this is our chance. We've got the experts in the room to clarify anything. Okay. Yes, Sue.
Susan Felter. So I'm wondering whether it would be helpful in the report to highlight some of the uncertainties around the dose metric in terms of part of what I really struggled with was the watts per kilogram. And, we heard today that body shape, it's not just mass, right? There are other considerations that impact the dose. And to ensure that there's not a tendency to just extrapolate findings based on kilogram body weight, which many people do in traditional toxicology and risk assessment, seems not to be appropriate here. And I think we'll talk about it more when we look at the biology tomorrow, but I'm not so sure that the differences between the rats and mice aren't also a reflection of body surface area in a way that it's going to be around twofold different or so, that they could explain some of the differences. And the fact that the males were the largest, what I keep seeing reported is that the male rats were more sensitive. They were exposed at the highest levels, right? And so if you think about any of us in contact with a wireless device, the bigger person's going to have a smaller exposure. And so I've already seen things, bigger males are more sensitive, but I don't think that would hold true for interpretation for what this, how you would think about it for human exposure. So I think that's going to be a tricky one in the report, and in terms of writing up the exposures, I just think that there's still more uncertainties about how the dose metric should be expressed. And a lot of caution is needed in terms of extrapolating it.
Thank you. Any of the technical staff or NTP staff want to comment on that or have anything else to add for the panel's discussion? Dr. Barnes, go ahead.
I'll just comment a little bit along those lines. The specific value of the SAR is that it takes into account the fact that different shapes you reflect different amounts, you get different absorption. And so in some sense it takes care of that by localizing what that absorption rate is at a particular place and time. For example, they showed the ears and the tail had a lot higher absorption than other places, and that takes into account the geometry of some of that. So it's there in that measure. Now when you translate that to, say, temperature change or some other change in the chemistry, that's a different matter. Then those things are different at different places and so on, even with the same SAR. So that's part, I think the SAR normalizes part of the problem. It doesn't solve the whole problem.
Can I ask a follow-up? Just make sure I'm understanding this. So if you took a 600-gram rat and a 300-gram rat, and you exposed them to the same watts per kilogram, say the 6 watts per kilogram at the high dose, would the exposure to the heart be 2 times higher in the males? Would it be equivalent in the males, or do we not know?
Well, it depends on what you're talking about. If you use the same calculation to go from the whole body weight to the actual, take into account the geometry and the various blood distribution and everything else, and you say what the SAR is at the heart, if you say those numbers are the same, then you come out with supposedly the same starting point for the effects on the heart.
Yeah, the goal was to keep all the exposure of all the tissue the same for the same exposure level. For rats, for mice, for different weights. So that was the objective. You don't completely achieve it because you have too many interdependencies, but that was the best. We achieved the best what is possible to do that.
And I applaud you guys for an incredible amount of work on this. I think that's my point, is that it's a lot more complicated than somebody looking at a summary table and looking at these doses. That it's a lot more complicated than that, and I think that really needs to come out in the report so that it's not misused.
Dr. Lin?
Yeah, I'd like to make a slightly different argument regarding going forward, if we might do so as a suggestion, whether one should focus on the 5G or try to learn more about what this current NTP program has produced. Of course, there's still a certain amount of unsettledness or uncertainty about the tumor development issue itself. Personally, I think it will be much more relevant to invest in a study in depth to explore whether what has been observed is something that will be repeatable, than jumping to another wagon to see if we can learn something about this new technologies that is coming on the horizon. That is not to say that that's not important. I think from a scientific point of view, if we can learn more about it, it's better than trying to just jump over to another area whereby it happened to be more fashionable.
Thank you. Our colleague from the FDA, yes.
Maybe I could.
Yeah, if you could do that, thank you.
Oh, this one is always on. I mean, now we're going to be moving from the technical aspects of the study design into interpretation of the data. And I think that a crucial element is to try to bring a little bit of clarity about what could be thermal effects and non-thermal effects. Because, I mean, some of the narrative that I'm sensing at this stage is that it has been shown that at this level of exposures that we're not dealing with thermal effects. Perhaps that's correct at the surface of the body. But we don't really know what temperatures the heart of these animals reached upon exposure. And we're going to be dealing with lesions of the heart. So I think that we need to be very cautious about claiming that effects that were observed in the study were not due to thermal effects but to some other mechanisms.
Would you introduce yourself, please?
Oh, I'm sorry. I'm Gonçalo Gamboa, Food and Drug Administration.
Thank you for that comment. I had a similar thought, especially when we get into the biology of energy localized to insulated nerve sheaths, for example. The biology there may be, in fact, very different than other parts of the body, which could be lending to some degree the specificity of what seems to have happened. And I just don't think we know enough to rule out either a thermal effect or some other secondary effect that's somewhat tissue specific. Dr. Barnes?
I'd like to comment on a pet peeve of mine, which is defining non-thermal. I think the only time you have a non-thermal thing is if you have one state or one particle. Otherwise, you're using temperature as a way of describing a distribution function. And so in my book, I haven't seen any non-thermal experiments. Now, you can say that additionally, chemistry is exponential in temperature, and typically we wind up with competing temperature effects, one going up and one going down as you change temperature, and so we operate in a relatively small region because you've got two chemical reactions stabilizing the system. And so I think looking at modulated temperature, and when you modulate the power, you modulate the input energy, and that automatically goes with the square of the electric field, and you're going to get modulation and demodulation effects. Now, they'll be small, but they go into an exponent of a chemical reaction. So I think dismissing thermal and modulated thermal is a mistake, as a place to start.
Thank you for that.
I want to add one thing. So, speaking as a non-biologist, take my comments with a grain of thought or salt. Just addressing the issue of being more forward-looking, looking at future technologies. My background initially was in EMC, electromagnetic compatibility, where first problems were IBM started having a computer and it would interfere with Aunt Matilda's television reception across the street. And gradually, a problem is found, the standard is changed. A new problem is found, the standard is changed. Even here, we're still in the same boat. We're realizing the entire world is changing far too fast for us to just react. But in the process of trying to predict, it's a very, very expensive task trying to go, okay, what's the next issue? What might we see? So I have a feeling that no matter how hard we want to go into doing predictive, mostly we're going to wind up being reactive for many, many years to come. And I also wanted to answer one question real quick. When NIST did do the measurements, we used water bottles, but they were filled with a simulating liquid. So I just want to come back to a point I made earlier, is the Italian study versus this study. Can you hear me now? Okay. There's a 60-fold difference in the high dose between the two studies with a very similar incidence of heart schwannomas in males. That's a dramatic difference in exposure. And if dose drives heat, there should be a dramatic difference in heat. So there's a lot we don't understand about the biology that's driving this. And before you talk about doing a study with 5G, try to understand the dose response here better with the data you already have and some refinement experiments on trying to understand what's going on in the specific cell types that are affected.
Just to clarify, I wasn't sure I heard what you said. When you said 60-fold difference for the NTP study, or talking about some other study?
So the SAR in the Italian study is high dose is 0.1,
yeah, okay.
and 6 in
that is correct. You were talking about the Italian study
Yeah.
versus the NTP
Yeah.
study. Okay, I understand.
And so just because I've not read that study, the Italian study, other than the abstract, is it fair to make that dose comparison? Were they similar exposures, and is that dose comparison correct between those two studies?
No, I think one has to be more cautious because the way it was done was entirely different from the NTP one. The dose calculations, they haven't provided much. In fact, there's nowhere close to what the NTP study has done. So there are a lot of reasons why you cannot just simply take that and translate it into what NTP has produced. Okay, it's entirely different experiment, actually.
I think that will be very important to clarify in this report, since the Ramazzini study is out, in terms of just making sure that people are doing appropriate comparisons with the uncertainties that are about that. Dr. Davis?
So I would actually remind everybody that we're we're really focusing on trying to peer review this particular study. And comparisons with other studies are probably not the best thing to do at the moment.
I think that the whole issue of dose just needs to be clear.
Exactly, and I understand that the investigators there had NIST people come to the site and review the methods that they were using. Thank you. The NIST investigators came and visited Ramazzini, and I also know that the Ramazzini pathologists have had some exchange, I believe, here with the NTP or with NIEHS pathologists. Is that correct, Dr. Bucher?
Well, again, we're here to review this study. We have had a long-standing relationship with the Ramazzini Institute to coordinate pathological diagnoses in studies, and I would say, yes, we've looked at some of their slides.
But in terms of exposure, they did have NIST scientists validate their exposure system, which obviously is not as sophisticated as the one developed by NIEHS, but was considered a valid system for their purposes. And yes, the exposures were, as you say, 60-fold and lower than those, and I tried to indicate that in one of my graphs, but may not have been clear. We'll talk about that more tomorrow.
Yeah, okay. Dr. Lin?
Yeah. I happen to have read that particular article, so I paid some attention to what was done in that particular case. As I said, the exposure system was entirely different. Of course, that's easy to tell. You just look at the two pictures, you'll see that's different. But how they calculated those was very, very different. In fact, what they really did was to use a formula and then say how much is exposure there is. Then based on the formula, they came up with a dose. This is what was done. It's not based on what was calculated for the animal that's being exposed or even tested with a phantom, even though I don't like your wrong phantom. But still, the level is very different. Understand they do also say that NIST actually had provided that formula, but that formula is not the internal to the animal-based type of calculation, but just external exposure translated via a formula that was used if you have a certain object, might be a sphere, might be a bottle, how much you might expect. That's what it was. It was predicted. Okay.
So I would just come back and echo Dr. Bucher's point that what we have to be clear on in this report for this study is to make sure that it is clearly described how the dose is measured and what the doses are in units, just so, because other people will take these other studies and compare whether it's the Ramazzini study or any future studies coming down the road. So I think that the dose issue is very important, and I think we've had a pretty thorough discussion of that today. Any other comments from any of the panel, questions from the panel to any of our experts? Seeing none, we're adjourned.